A pleasant morning to you all. Honorable Chief Guest, Professor Duburu Jamuna Madam, Vice Chancellor of Sri Padmavati Mahila Vishwavidyalayam. Our Honorable Guest, Professor D. Mamata Madam, Registrar of the University, and guest, Professor R. Nagaraju Sir, Dean, School of Sciences, and all our other guests, welcome you all. My name is Dr. G. Sirisha, Assistant Professor, Department of Home Science. It's my immense pleasure to welcome you all to the today's 2 days international virtual conference on nutraceuticals and functional foods for human health 2022. We are grateful to our organization, higher officials, and organizing committee under all the volunteers who have supported to organize such a beautiful program. I wholeheartedly welcome all the delegates across the country and outside the country. My wholehearted welcome to today's keynote speaker, Dr. Srinivasa Janaswamy sir from South Dakota State University, USA. I extend my welcome to our distinguished speakers, Sri Naveen Kumar Rastogi sir from CFTRI Mysore, Dr. Pushpa Bharati Madam from Dharwad, Dr. Manjula Madam from SV University, Dr. Rekha Sushil Kumar Sharma from Nagapur, Dr. Hula Juhu from Amrits. Welcome you all for the today's conference. The theme of this conference, nutraceuticals and functional food for human health. The main aim of the conference is to promote latest research advancements in food technology and its related fields and sharing the new products in the field of nutraceuticals and functional foods, that is on prebiotics, probiotics, other food compounds with potential health benefits and its specific nutrient effects. Mainly, nutraceuticals and functional foods provide an opportunity to improve the health, reduce healthcare cost, and support economic development in communities. That tend let food be the medicine and medicine be the food, espoused by Hippocrates nearly 25 years ago, is revising renewed interest. Now there has been explosion of interest in the health enhancing role of the specific foods or physiologically active food compounds and components, so-called functional foods. So while nutraceuticals sometimes referred to as natural health products are often used in medicinal forms as tablets, capsules, or in the form of liquids also. So nutraceuticals and functional foods for human health is more suitable trending research. Because of this, we are organizing the present conference. So all of you, please utilize the opportunity. Thank you very much for your active listening. Let us welcome Professor M. Aruna Madam, Head of the Department from Home Science Department, Sri Padmavati Mahila Vishwavidyalayam, to share few words about the university and the department. Now I welcome you, ma'am.
Thank you, Sirisha. And good morning, everyone. Good morning to the distinguished chief guests and also the Honorable Vice Chancellor, Professor Duvur Jamuna Madam Garu, and Registrar Professor Mamata Madam Garu, and also the distinguished guests from Dharwad University and CFTRI, and also from other countries. I welcome you all for this program. And it's my pleasure to have an association with the Department of Home Science as head of the department. And I thank Dr. G. Sirisha for organizing this international webinar. And I welcome you all once again for this program. I also welcome the students and also the participants of various places. Now, I would like to share about our university, because our department is one of the biggest department in the university. And about the university, Sri Padmavati Mahila Vishwavidyalayam, established during the year 1983-84 as a state university for women with a motto to empower women and train them to be economically productive. So currently the university offers 58 postgraduate and undergraduate programs to 6,400 plus girl students and the women, with the help of 96 regular academic staff and 159 contract and academic consultants. The research programs like doctoral research programs for around 200 national and international women students is being offered in different departments of Sri Padmavati Mahila Vishwavidyalayam. 90% of the academic programs offered by SPMVV are applied and also career oriented. I would like to mention few words about our university, and the university has achieved the credit of running technology business incubators and also Rural Women Technology Park, TEP Outreach Cluster Innovation Center, funded by the DSIR and PRISM, and Institution and Innovation Council Skill Development Center. And we also have DST Curie Artificial Intelligence Center, and BioNest, and United Nations Asian and Pacific Training Center for Information and Communication Technology has identified Sri Padmavati Mahila Vishwavidyalayam as the Training of Trainers TOT Center for Women and ICT Frontier Initiatives Wi-Fi. This is the only center that is being established in India. And about our department, I would like to say a few words about our department, because our department is also established in the year 1983, almost 39 years from the inception. This department was existing. With the main objectives of the department is to offer career-oriented undergraduate and postgraduate programs, and to provide research experience through MPhil and doctoral programs. And we also, to our credit, we also have some research projects, and to involve in extension and community outreach programs, and also the activities that will be taking place regularly according to the important days that were being organized. And it also provides an hands-on experience through internship, workshops, and training programs. At present, our department is offering different programs like M.Sc Clinical Nutrition and Dietetics, M.Sc Community Health and Nutrition, and M.Sc Food Science and Quality Control. These are the three specializations which will come under Food and Nutritional Sciences program. And recently we also started, that means in the year 2014, we have started M.Sc Integrated 5-year M.Sc Integrated Food Technology program, and under which this international webinar is being organized. And we also have initiated from the 2014 itself, we have started 2 Bachelor of Vocational Degree programs, which was sanctioned by the UGC under skill development programs. Those are Bachelor of Vocational Degree in Nutrition and Healthcare Sciences, and we also have Bachelor of Degree in Fashion Technology and Apparel Designing. And it is to our regarding the research programs, we also have almost 67 PhDs enrolled under our department, under various staff. And recently we have seen that many of our senior staff have been superannuated from their services. So because of that, there is a lacuna that is being falling ahead to take many of the research candidates. But of course, we all have very good strengthened staff who can motivate and who can guide the students. And we also have mentor-mentee system, which is very strong in our department. And we, as a food scientist, we will work in various thirst areas like food science and nutrition, food technology, therapeutic and nutrition and healthcare, and which is also aims in training the students in various aspects related to food processing, which is one of the nurtured subject of interest in our department. And the students experimentally, practically by learning and developing various products, therapeutic products, as well as a new technological technology processed products that have been developed in our department. And to our credit, we also will be reaching to the community often. And Women's University will create a platform to train the youth and also the students to empower them regarding their entrepreneurial orientation and also start their own enterprise. So the university and department have a strong base with related to the dimensions of research and also the outreach and entrepreneurship as well. With this, today's international webinar, mostly we have concentrated on the nutraceuticals and functional foods, which are being used often in the health and in the prevention of health and disease. So Greek physicians as well as the Hippocrates, known as the father of medicine, and several years back they have mentioned that let thy food be medicine and medicine be your food. So the philosophy behind is mainly focus on prevention. So prevention is better than cure. So nutraceuticals as well as the dietary supplements, whatever the terminology that is being used, the functional foods, etc., are the main terminology that is being used to prevent the disease, various diseases. So the term nutraceutical has been used to describe a broad list of produce sold under the promise of being dietary supplement, that is as a food, but for the expressed intent of treatment to prevent the disease. So a foodstuff is being fortified and food or dietary supplement, which is also going to provide the health benefits to the individuals. So the functional food concept is also being defined as food products to be taken as part of the usual diet in order to have beneficial effects that go beyond basic nutritional function. So the nutraceuticals are present in most of the food ingredients with varying concentrations, and also the concentration, time, and duration of supply of these nutraceuticals influence the human health. So the manipulation of the foods, the concentration of active ingredients, which can be increased in order to benefit to the majority of the populations. So the diets rich in nutraceuticals, along with regular exercises, stress reduction, and maintenance of healthy body weight, will maximize the health and reduce the disease risks. So with this intention, the present webinar has been organized by Dr. G. Sirisha. I hope that this will give a fruitful and rich experience for all of you. Thank you very much.
Thank you, madam. You explained very well about the institute and our department activities, and also about the seminar. Thank you so much, ma'am. Good morning, sir. Our dean sir is joined, madam.
Okay.
Good morning, Nagaraj garu.
I welcome Jyoti, ma'am.
Thank you, Sirisha.
How are you?
Fine, fine, sir.
Okay, good,
How are
good.
you?
Yeah, very nice.
Thank you, thank you. Registrar madam vachinattunaru.
Yes, Andy. I'm in.
Good morning.
Madam, good morning. Sir, good morning, sir.
Good morning, madam. Now I invite Sujata introduce about our registrar, ma'am.
A very good morning, dear participants, invited guests, distinguished speakers, dean sir, reputed professors, and our beloved registrar madam. A warm good morning, everyone. I thank Sirisha for giving me this opportunity to speak, to introduce our prolific researcher on campus, Professor D.M. Mamata madam. She is also registrar of our campus. Can I request her? Thank you. The biodata which we are presenting is a very, very small and a brief one of her research work. But I just want to mention that she has been an active person behind establishing various incubation centers and promotion of research-related activities on campus. It's my immense pleasure to introduce this person in this event. Madam has a wide research expertise in biotechnology, molecular biology, and she's well known for her gene cloning work throughout India and also across globe. She is a proud recipient of Fulbright Fellowship in the Department of Entomology, University of California, Davis. She served there from 2013 to 14. She has also been the Oversight Young Scientist Fellowship at the same department, which was funded by DBT during the period of 2007 and 8. Madam has numerous projects which she has already completed and which she is still handling. The number of projects she has handled are very, very huge, and their major funding sources are from DBT, UGC, DBT, Indo-US, and Indo-Egypt collaborations. Madam has been recognized for her extensive research activity, and she has received various honors and awards in this context. She is UGC International Travel Awardee. She is National Academy Summer Fellowship Awardee at Indian Institute of Science, Bangalore. 2 times. She is a lifetime awardee of International Society of Ecological Communications, and she is also a DBT Young Scientist Fellowship Award, Overseas Young Scientist Fellowship, which was awarded by DBT, Government of India. Madam has also received WHO UNICEF Achievement Award at Bangkok, Thailand during October 2006. This is a very, very brief introduction about Registrar Madam. Thank you, Madam, for accepting our invitation and gracing this occasion. Now I request Registrar Madam to share her thoughts in this event. Thank you, Madam.
Thank you, Dr. Sujata. Thank you for your kind words, which I really don't deserve. But thank you,
Madam, you deserve it.
Given me. Yeah, good morning, everyone. The August audience of the 2-day International Virtual Conference on Nutraceuticals and Functional Foods for Human Health. Let me greet each and everyone, the Dean of School of Sciences, Professor Nagaraj Garu, the former Dean and a dear Professor Jyoti, and the esteemed faculty members from the Department of Home Science, and the invitees and today's speakers. A very warm welcome and morning to you all. Well, the very theme of food science always fascinates me because this is one of the subjects which I always cherish because it's taught me what to eat and what not to eat, and how much to eat, and what is that I'm eating, and how much my body requires. And sufficient training, I'm glad to share that sufficient training has been given by my teacher, Professor Jyoti, who was also here in this conference. I acknowledge her, but she made me learn how to make the diet sheet over and over during my studenthood days, which gave me enough exercise and also a knowledge and how I'm supposed to be, in maintaining an ideal body weight, and also the foods that I'm supposed to take. Well, about the theme, it is said, you are what to eat. Explains and tells what you eat, and I will tell you what you are. Well, the nutraceuticals or the functional foods are the foods that provide both health benefits to reduce the risk of chronic diseases and basic nutrition. DFLS in 2002 defined nutraceuticals as a food that provides medical or health benefits, including the prevention or the treatment of the disease. It is when a functional food aids in the prevention or the treatment of diseases or disorders other than anemia. It is called as nutraceuticals. You all must be knowing. But today, nutraceutical is a term commonly used in marketing to describe any product derived from food source that provides extra health benefits in addition to the basic nutrition value that is found in the foods. Such products typically will claim to prevent chronic diseases, improve health, delay the aging process, and also increase the life expectancy. Well, on the way, functional foods also provide important nutrients that can help protect against diseases. Many are especially rich in antioxidants. These molecules help neutralize harmful compounds known as the free radicals, helping prevent cell damage, certain chronic conditions, including heart disease, cancer, diabetes, and other diseased conditions. The changing eating habits, actual trends in production and consumption have a health and environmental and social impact too. The European Union is fighting diseases characteristic of modern age such as obesity, osteoporosis, cancer, diabetes, allergies, and dental problems, while the developed countries are also faced with problems relating to aging populations, high energy foods, and unbalanced diets. The potential of nutraceuticals, functional foods, food supplements in mitigating health problems, especially in gastrointestinal tract, is very important in the current context. Certain members of gut microflora in the gut, also we call it as probiotic or protective strains, play a vital role in the host health due to its involvement in nutritional, immunologic, and physiological functions. The potential mechanism by which these nutraceuticals or the functional foods or also called the food supplements may alter a host health are also of utmost importance in today's context. An establishment of novel functional cell models of the GI and analytical tools that allow test in controlled experiments are highly desired in the area of research of nutraceuticals and functional foods. And therefore, the nutraceuticals or the functional foods are also called the food supplements, including the probiotic, prebiotics, symbiotics, phytochemicals, especially the need for consuming appropriate diets, health issues are also play a key role in keeping up good health. Overcoming the failure to adhere to the known healthy eating models, development of new nutraceuticals or the functional or the food supplements with novel health benefits, elucidation mechanisms of action of these products, development of study systems such as in vivo co-culture cell models are also of great priority in the current context of pandemic too. Appropriate diet culminates in a healthy, properly functioning GI tract, resulting in attainment of proper human physiology and hence healthy living too. Otherwise, the opposite becomes true. So modeling new eating habits using the existing knowledge is needed for an eventual ideal of health for long vision. In this context, the today's resource persons, Cinnamon Swami from Faculty of South Dakota State University, who has consented, graciously accepted to be our speaker and also to address all our participants. I really thank him for taking time to be with us among us. And also I thank Naveen Rastogi from CSR and TI, who has also agreed to speak to all of us on this platform and on this theme. So I wish all the participants, students, scholars, and faculty members a very fruitful time and listening to these leading lectures. I wish you all a very great time and a fruitful time. And on this occasion, I also congratulate the organizing secretary and all the faculty members from the Department of Home Science who made it possible to happen through our university. Thank you, one and all.
Thank you, madam. You explained very well about the nutraceuticals and functional foods and its health benefits. Thank you, ma'am. You are the backbone for the today's conference. Thank you so much for your support. Now I welcome Srinivasa Janaswamy sir. Good morning, sir. Welcome you for this conference. Now I welcome Indira Priyadarshini ma'am to introduce our Dean, sir.
Thank you, Sirisha. Beloved Registrar Madam, Dean Sir, and all the invitees and organizers and participants of today's webinar. Very, very good morning. And it is my pleasure to introduce our dynamic Dean, School of Sciences. Sir has obtained B.Pharm, M.Pharm and PhD degrees from Manipal College of Pharmaceutical Sciences. And Sir started his career in Vishweshwara Institute of Pharmaceutical Sciences. Later on, he joined in Sri Padmavati Mahila University in 2004 as professor. He opened the windows of newer ideas, and he started M.Pharmacy Pharmaceutics course. And Sir is presently working as Dean, School of Sciences, and Director of Research at Sri Padmavati Mahila Vishwavidyalaya. He has successfully raised project grants of about 2.37 crores from DST, UGC, and AICTE. Professor has been granted 2 patents on dental implants. He organized UGC refresher courses and national seminars and international seminars, and he has delivered more than 100 lectures in various national and international seminars. And our professor has published more than 75 research papers in national and international journals, and he has guided about 46 PG students and 12 PhD students and 8 presently on rolls. Dr. Nagaraj Garu is research and a research consultant to various organizations and has been member of governing council of few educational institutions. He has been editorial board member in reputed journals and board of studies member in various universities. Sir held many administrative positions in SPMVV as head director, Center for University Ranking, deputy director, Directorate of Distance Education, chairperson IAEC, Dean Development, etc., and now as Dean. He received many awards to his credit. Best Pharmacy Teacher Award by Association of Pharmaceutical Teachers of India in 2018. State Best Teacher Award 2017 from Government of Andhra Pradesh. He received Pharmaceutical Scientist of the Year Award 2017 and Best Teaching Pharmacist Award, National Level Pharmacist Award, and Scientific and Innovation Research Award in 2017, a National Best Teacher Award in 2017, and Distinguished Alumna Award of Manipur College of Pharmaceutical Sciences from Manipal College, and recipient of Achiever of the Year Award, AVOPA Bangalore. This is very, very brief biodata of our Dean, sir. And I thank Sirisha for giving me this opportunity. Thank you, Sirisha.
Thank you, madam, for your nice words about Dean, sir. Sir, now I welcome you for say a few words about today's conference. Sir, you are in mute, sir.
Yeah, how is?
Yes, sir. Now it is audible.
Good morning, respected Registrar Madam, and our senior colleague, Professor Jyoti Madam, former Dean, and other colleagues of Department of Home Science, and the invited speakers, and also the most important, the delegates of the today's seminar. And I should congratulate the department for having a very apt topic for the pandemic time, where the whole world is suffering, and you are really come out with a nice title and to speak about it, which is the actually very important in case of how we are going to go with this in this condition, that is functional foods and nutraceuticals. So it was very interesting to me as a basically I'm a pharmacist, and we do a lot I should say the competition from nutraceuticals with the pharmaceuticals. And anyway, we wholeheartedly support the field of nutraceuticals and also the functional foods as medicines in mitigation and the relief of the disease conditions. And the food, the functional foods and nutraceuticals are not new to the Indian society. When you see our Ayurveda and such other Vedas, the food, they said, is very important. The diet is very important. It is also called as patyam. That means what you are supposed to take and what you are not supposed to take. And it is also mentioned 2005 years, 2,500 years ago by Hippocrates. Let the food be thy medicine and the medicine be thy food. So it is very important, and food also provide therapeutic benefits, which is now the duty of all of us to establish what type of the foods and what type of benefits, because now the modern society requires the evidence. What evidence you are going to say this? So that's what the whole gamut of functional foods and nutraceuticals is going on. And however, the food as a medicine has taken a backseat when you see the history, when you see the past in 19th century, where we have, as a pharmacy, a modern drug therapy has overshadowed the whole food as medicine concept. But fortunately, after 1900, the importance of the diet and the in the disease prevention and health promotion came to forefront again. That's why this field has picked up again in the last few decades, and really doing very good. And a lot of inputs, a lot of research is required here. And as I said, to make it more affordable and acceptable for the people of the country, especially when you are talking about India, where the literacy rate is very less. And I'm not going to details of the definitions of the functional foods and types of functional foods. But anyway, the functional foods are generally, all of it is conventional and modified foods. So the conventional foods basically fall into the category of fruits, vegetables, nuts and seeds, whereas the modified functional foods are the juices, dairy products such as milk and yogurt, and grains and bread and pasta and fortified eggs. And all these, when you see the literature, the nutraceuticals are often synonymously mentioned as functional foods also in the media and also in some of the part of the literature. But in fact, the nutraceutical is different. It is the term coined by Stephen D. Phillips, founder of Foundation of Innovation in Medicine and Medicine in Concord, New Jersey. So the nutraceuticals have got the correct definition now as a parts of the food or the whole food that provides medical or health benefits, including prevention and treatment of diseases. So therefore, as I said in the beginning of my speech, it is going to be what I should say, the competition for the drug industry. And the normal food, if you're able to control the disease condition and mitigate the disease condition, then what is the purpose of going for the expensive drugs, exposing ourselves to the toxic effects of the drugs? So that's why when you see the market abroad, the developed countries, there is a lot of nutraceutical market is available. And I have little statistics of that, and where in India, the nutraceutical market is yet to pick up, and probably we require more evidence-based research in bringing out something which we wanted to bring into the people of India. And when you talk about the market scenario, it is very difficult for any company to cater the food needs of the market. So we have a highly diversified socio-cultural background and regional prosperity and education. So therefore, we can't just make it a particular functional food or particular food item or particular food which is going to be acceptable the whole length and breadth of the country. So therefore, that is the main reason when you just come to the south, we have different food habits. When you go to the north, we have the different food habits. When you go to Tamil, even in the south, in the Andhra, and we have Kerala, we have various states which are having their own food habits and the types of the foods what they are going to take. So that is a major challenge for the home science department or the scientists working on nutraceuticals to bring out which is going to be acceptable in the market point of view to all the people. Therefore, we will be able to minimize the use of the drugs, toxic drugs, and also making the effective and economical treatment compared to that of the drug therapy. So the demand, the US market, when you talk about the food processing, the US market is we have around, as for my statistics, I am subject to corrections. The Home Science Department, pardon me if the statistics are wrong. So it is going to have almost nearing 1,000 billion US dollars in 2025, and it is going to make it 1.7 trillion by 2026. So here, that is a potential market we have, and if any of our research and our innovation in this angle will, the nutraceutical and functional foods probably will have a great market value. That does not mean that our country policy makers are sitting quiet. The policy makers are doing a lot to the people so that to make them what I should say, very good supply of the food. So we see the statistics say that 15% of our Indian population are undernourished. So then the government is taking a lot of several measures to reduce these undernourished people through the initiatives such as Integrated Child Development Services, National Health Mission, Midday Meal Scheme, and several such other schemes where the children, undernourished children, will be provided with the basic food, which is going to be very, very important to us. So the strength of the Indian nutraceutical market is also emanates from certain supply drivers, which place it among the strongest market in the world. So it is I should congratulate the Home Science Department thinking of a topic that in the pandemic where we see the immunity plays a very important, and also the food, what type of the food you are taking to be your face the pandemic situation during infection or before infection of the corona or so on. The food plays very, very important. So this is a very apt, and I see the renowned speakers like Cinnamon Swami Garu and other invited speakers were taking out your valuable time and spending with us, with our university, for the sake of our students is well appreciated, sir. As our registrar madam always says, such thought of spending time with our people like you will be a long way to go for enlightening our students about what is happening in the field of respective field. What is your specialist or invited to talk about it. So in this with these few words, I congratulate and wish the participants. I congratulate the department and wish all the participants to make the best use of it. You are the best speakers around, and even though it is online, it is no problem. We are used for last 1 year, and that's why we could connect the people across the globe. Otherwise, it would have been very difficult for us to bring the people from America or some other part of it to speak to our students. Would have become an impossible task for universities like us, and also the money involved and the time. So this is made a good platform for all of us to interact with the specialists like you. So with these few words, I appreciate the department and also thank the organizers for giving me this opportunity to be part of this a good program. Actually, I wanted to sit through the program, but due to some administrative emergencies, I need to leave. Please kindly excuse me. I will be leaving after 5 minutes. And thank you, Professor Sirisha, for giving me this opportunity and share your thoughts of mine.
Thank you, sir. Our dean sir narrated very well about nutraceuticals and functional foods in pharmacy terms and also in nutritional terms also. And its importance in present pandemic situation and market of these functional foods and nutraceuticals, sir said very well. Thank you, sir, for your few words. Now I welcome Madhavi, Dr. Madhavi, to introduce today's speaker, Srinivasa Janaswamy, sir. Welcome you, Madhavi.
A warm good morning and welcome to all the delegates of today's conference. It is a great honor to me to introduce today's keynote speaker, Dr. Cinnamon Swami. Currently, he is working as a faculty member at the Department of Dairy and Food Science, South Dakota State University, Brookings, USA. In 1997, he awarded doctoral degree on crystallography from one of the esteemed educational institutions in India, that is Indian Institute of Technology, Chennai. From Mysore University, he received MS. Specialization was Physics and Education. Bachelor's degree was Physics, Mathematics and Electronics from Nagarjuna University, India. Dr. Janaswamy sir has international research collaborations with groups in China, Kenya and India, and visiting professor appointed at the high-end foreign expert state key laboratory of Food Science and Technology at Jiangnan University, China. School of Food Science and Technology, Dalian Polytechnic University, Dalian, China, and also in India, Vellore Institute of Technology, Vellore. Formerly, sir served as research associate and assistant professor in Department of Food Science, Ford University in USA. He has more than 25 years of research experience that constitutes an interdisciplinary at the frontier between carbohydrate chemistry, physics, material science, and life sciences. He leads a research program on functional carbohydrates with an emphasis on the design and development of novel carriers of bioactive compounds. His research also focuses on agricultural residues towards packaging and biodegradable materials and water purification. Over the years, Dr. Janaswamy's research gained high-profile media exposure through journal editors and scientific writers' press reports. Several scientific news agencies around the world covered Dr. Janaswamy's research and the encapsulation and delivery of health-promoting and disease-preventing compounds through polysaccharide fibers and starch matrices and also cellulose-based biodegradable films. He has 7 patents in his research career. He published around 85 international peer-reviewed journal articles so far, edited 1 book on natural polymers for drug delivery, and edited 1 special journal issue on green physical processing technology for the improvement of food quality. Currently, he is serving as an editorial board member of Food Chemistry. Food Chemistry Advances, Functional Food Science and Physical Chemistry and Chemical Physics. More recently, Sir was received the 2019 Global Engagement Research Award under 2022 David Fee Memorial Lecture Award from South Dakota State University. Based on this, he is very active in research and academic work. I am very glad to welcome Dr. Jeneswamy Sir, invited keynote talk on bioactive compounds as sustainable tools towards developing novel functional foods. So I think it is a very relevant and useful topic for us and also to our university students. Thank you for giving this great opportunity to the team of this conference. Thank you. I am welcoming you, sir.
Sir, you are on mute, sir.
You have to unmute.
Okay, now you are unmuted, sir.
Can you allow me to share my screen, please? Yep, thank you. I think I am. You are able to see my screen?
Yes, sir, we are able to see your screen.
Can you hear me?
Yes, sir, you are clear, sir.
Okay. Thank you very much. As Dr. Duvuru said, all my experience, basic education was in physics, but it could be quite interesting for you all how a physics person could come and talk about bioactive molecules. But believe me, for the last 25 years, I have been working in the area of carbohydrates. And my focus has been on looking at the three-dimensional structures and understanding the functionality of biopolymers. And then I am using that knowledge to develop these novel functional foods. That has been there, my dream for the last 10 years, to be frank with you. And especially we are trying to use how bioactive molecules could be used towards developing these functional foods. So my focus has been on developing the carriers of these systems, and parallelly we are developing the functional foods, some food products, but we are yet to venture into developing the functional food based on the carriers we developed. But I try to bring some linkage between these two, and then hope I will meet your expectations. So as my title says, I am bioactive molecules from the natural sources as a sustainable tools towards developing functional foods. As Dr. Nagarajan said, the functional food is a buzzword right now, right? Because as we all know, we have varieties of compounds that could be extracted or envisioned from a plethora of sources. For example, vegetables we could use, and also fruits, and in grains, oils, whatnot. There are various of sources that we could extract these health promoting and disease preventing compounds. And for a long time, their effects have been known. For example, ginger, for example, in a layman term, if you want to say, we all know that it helps with the digestion. It improves the digestion. And same way, the Caspasian and the chilies. And then also the turmeric powder that we use in our as a spices, and that is predominantly the curcumin, that aids to address the diabetic issues and whatnot, varieties of other issues as well. And I am not really sure how many are fond of eating grapes. And then when I was a kid, I used to eat grapes, spit all the skin and then seeds. I used to eat only the pulp. But now only I realize that all the health promoting compounds are there in the seed and the outer layer. The seeds, though they are bitter, they contain a bioactive molecule called as resveratrol that helps with varieties of functionalities. Okay, and if you look at it, my statistics are a little different from that, Professor Nagarajan's statistics. I just found out that the global bioactive ingredients market, if you look at it in 2021, it was around $27 billion, not billion dollars, please remember that. And that is projected to reach around 50 billion by 2026. In 4 more years, I think already 22. So in 4 more years, it would hit 50, 51. This is only projection, but it could even go very well. So why? Oh, by the way, this market, when I'm showing it, is not only the nutraceutical compounds, but also other probiotics, prebiotics, and fiber and everything, whole gamut. you could imagine. All of them are incapacitated here, but one interesting thing here is the market trend is increasing. That is very important thing that for us to understand. Why? Mainly because the consumers are really, they have the knowledge now. I can tell you, maybe I sincerely believe the dietary guidelines that have been implemented, I think 1992 was the one that started first. So for the last 3 decades, whatever we have been doing in the society, educating the general public about health benefits of eating nutritious food. So what is best, what is not best. So I think that would have really helped, and then that would have educated the general public in understanding to be healthy. And then they got convinced now that their health is there in their food. Okay, so that is, and especially the younger generation now, if we speak, the college goers now, they are very much interested in having healthy food. They are not succumbing to the food that normally we used to gobble when we were kids. And then during this process, they don't even mind spending some extra money. That is how we have all these whole foods, and the new foods are coming up, and this functional food market is also driving. These are some of the sources. So, and also more importantly, when, I don't know, you all would have noticed this, because I don't know how many of you are reading the label, but that is the new trend. So nobody is just like that grabbing the food that they get from the shelf. They're clearly seeing how many calories that food contributes, and what are the calorie contributors. Are they coming predominantly from the carbohydrates, or are they coming from the protein or from the fat? And then we all know the effect of salt, so we want to reduce the salt in the food. So they are looking at how much of salt that food contributes. And then more importantly, nowadays, we are all interested in having a lot of fiber in the system. So they are even looking at the amount of fiber, dietary fiber that they could get. And furthermore, the amount of vitamins, and then the minerals, all that. So people are paying a lot of attention to the nutrition label, which is a good thing. And more importantly, as this lady is asking, they wanted to understand how those foods are being processed, where they are being grown. If they are vegetables, are they grown on with pesticides, or how are they being stored? So all these questions people are asking, and this in a way contributes to this the new change that we are all experiencing at this moment. And I mean, though these two are cartoons, I just wanted to emphasize that. And furthermore, the public is looking for the clean label, in the sense they don't want to hear something that is unknown to them, and they don't want to have a long list. So that's why she fainted here when she listed the long list, because they won't have a concise list. At the same time, we all know that unfamiliar names are not at all acceptable. For example, starch is nothing but alpha-D glucopyranose. It is connected to the 1,4 linkage and 1,6 linkage. So if you say in the label that 1,4 linkage alpha-D glucopyranose, boom, we will be scaring the consumer. So that is where we wanted to come up with a label names that are familiar with them: a flour, rice flour, corn flour, or chocolate, butter. Such type of things are. So these things indeed are going to put a lot of stress on the food companies, and especially when they are trying to add some bioactive that have different chemical names. And also, if you look at it, the trend for a long time, the consumers are dictating what they should have in their food or what they cannot have. For example, the craft, but for a long time, you all would have heard of. Okay, by the way, all these examples I chose from the US perspective, but I'm pretty sure they will be applicable for the Indian society as well. So the mac and cheese that people have been eating, predominantly for a long time, they have used artificial preservatives, the colors. And we all know that, because when you consume, there will be a stain on the tongue on our kids. We could see that. And then that even gets secreted out through urine and other ways. We could see that. But nowadays, the trend is now we don't want to have any artificial this synthetic compounds on those foods. And same way, and the Chipotle, that is also one more fast food restaurant, they are not going to add any GMOs. And then most importantly, I think for a long time, you all are familiar with the diet, diet Coke and all those things, drinks. All this sucrose has been replaced with a sweetener, aspartame, which has done a market for like 2 decades, I believe, more than 2 decades. But however, now we realize that there is a lot of research that shows that aspartame is not good because it could lead to cancer and other issues. So now we want to strip off those things. So people are demanding that we should not have any product that contains aspartame. So consequently, the companies like PepsiCo is listening, and then they are trying to come up with alternatives. How fast they could come, how easy, that is a matter of time. And same way, Nestle is removing all the artificial colorings, the M&Ms and Skittles, anything, any color balls that our kids eat. They all used to have artificial colorings. So artificial compounds, synthetic compounds. So we would like to get them off. And one more interesting thing here, you all might be aware of, that is the social media plays a significant role in this. I can tell you more recently it happened only a couple of 3 to 4 years back. We all know that carrageenan, a polysaccharide that is extracted from seaweed, is being used in food and pharmaceutical applications significantly. But however, all of a sudden, somebody felt discomfort in their stomach when they consumed a beverage that contains carrageenan. So she posted on the internet saying that, hey, I consumed this product. This has carrageenan, and then since consumption, I had some stomach upset. That went as a viral, I can guarantee you. Then after somebody replied saying that, hey, there is a research that shows that polyginan could induce cancer. Maybe this beverage is having issue. And then somebody even explained, though they are not scientists, they said, yeah, in our tummy as also there are some harsh acids that could break down this long chain polymer into oligosaccharides, and that could induce all this issue, which is not true, because it will not happen like that. All these polysaccharides, these high molecular polysaccharides, they get secreted out. They work as a dietary fiber. But however, wrong news, it spreads like wildfire. We all know that. So, and thus what really happened was consumers stopped buying all the products that contains carrageenan. And then many food companies, they're trying to strip off all the products that contains carrageenan, but it is really challenging to come up with a product, a polysaccharide mixture of them, that could really do the same trick as the carrageenan does. So it is challenging too. But however, now a lot of research has been done, and then they proved that all those claims are wrong. And then now I think once again, carrageenan is getting back its claim. But what I wanted to emphasize here is the consumer is driving what they want to have in their food. So this really gives a challenging scenario. This leads to a challenging scenario to the food industry. And a couple of more examples I would like to share with you is, for example, one research that clearly shows that pigmented potatoes, I think many of you would have seen that purple potatoes, red potatoes, yellow potatoes, white potatoes. So if you look at the way the starch gets digested, that clearly shows that depending on the amount of polyphenols present, if you have more amount of polyphenols, there is a less starch digestion. There is a negative relationship in the sense. So the more amount of this polyphenols present, that reduces the overall starch digestion, which is really interesting. We all know that potato consumption is not good for health. That is a notion we have, which is wrong. But however, now if you could add this, the colored potatoes to your diet, it appears that could help to improve the health for sure. But here, this research was done predominantly, I think, this in vivo research only. But one more interesting thing that we had to learn from this exercise here is all those healthy compounds are on the outer skin. Okay, so we should not peel off the skin given a chance. If possible, consume the potatoes along with this the peels if possible, because that's where all the healthy compounds are hidden. And same way it happens with the rice too. Rice. All the polyphenols and the fiber, everything is nicely coated on the outer layer, which we are trying to strip off. And then during the polish process, and we end up having the pure white carbohydrates material, which is nothing but the junk starch. And then that consumption will onset other health issues, which we don't want. Okay, and one more interesting research this was done for about 24 years in about 125 US men and women. For a long time, our grandparents and our parents, they insist on saying, eat vegetables, because many of them are rich in flavonols, anthocyanins, flavonoid polymers, all these are healthy. So this study clearly tells us that by consuming those fruits and vegetables, that would help to maintain the weight. So this is also very interesting finding. So once again, it boils down to this bioactive molecules. That's what I can tell you. And then the curcumin has been proven for a long time that the curcumin turmeric extract, okay, that is a curcumin. They did a 9-month intervention study and the prebiotic population, and then that clearly shows the beneficial of curcumin in preventing that type 2 diabetes, which is really, I mean, some of these things are known, but however, now we have scientific evidence that clearly shows that. And there is one more I would like to share with you. This is the resveratrol, predominantly comes from the grapeseed is extract. That also, in the mice studies, that clearly shows that it could control or reduce the tumor growth, the lung cancer. So this would have convinced you all. I mean, I know this whole theme of this conference is about nutraceuticals and functional foods, but further, at least this would have reinforced your thought that, hey, these healthy compounds, the fruits and vegetables are rich in healthy compounds and their consumption is good for health. But however, as we all know, that some of these fruits and vegetables are not available throughout the year. And that's where if you could come up with a way to extract these healthy compounds and later add them to the food matrix, that would be challenging. And that is the focus of all of our efforts are driven in this direction. Okay. So, I mean, once again, so the varieties of functionalities, health benefits, they help to address the diabetes, cancer, cardiovascular disease, aging, asthma, hypertension, whatnot, anything I think we could find. But only thing is we should know which one to use, because there are wide varieties of bioactive molecules. Each one functionality could be different. Always remember that. Okay. So, and this food industry, now they have realized it looks like these bioactive molecules have to be included in their food. So they are trying to identify the foods that contain these bioactive molecules. And then they are spending a lot of their time and efforts in developing these modules and then come up with some of these nutritious foods, these functional foods. Already there are some in the market, but some more I think would come. But if you look at the patents, there are varieties of patents. I mean, this was done in 2015, the list I took, but still we could see by then itself there are many of the patents are coming up. All of them are filed by the companies only, not in individual groups, which is a good sign, to be frank with you. And then, as I said, the food industry is really focusing on it, and then hopefully down the lane we'll have all these functional foods. But now, what are the couple of challenges that we all face when you're trying to extract these bioactive compounds? Because now we got convinced there is enough evidence that consumption of these foods will be healthy. But okay, now how can we extract them? Because it will be challenging, to be frank with you, mainly because these compounds, they don't really exist as a separate molecule. It's not a free molecule. What really happens is they conjugate, and they covalently link to the matrix. It could be cellulose matrix, hemicellulose matrix, lignin, or anything. Somewhere in that matrix, they're nicely attached, but they're covalently linked. So during that process, extracting them is quite challenging. And also, for example, one more thing, we all know that there will be year-to-year variability. So always, let us say you are extracting some compound from the pear peel, the pear skin, you may not get the same amount from every year, and also from location to location, there will be dependence. I'll show you one of my research that there is a location dependence in the anthocyanin amount. Same thing happens here. So these are some of the challenges one has to face. But one more thing is there's stability, because many of them are not stable. They're not water soluble. So we have to use appropriate solvents, and many of them are temperature unstable, and then they get oxidized the moment they expose to oxygen, and then pH plays a major role. So, and consequently, the extraction is a challenge, and once you extract, also adding them to food product is also a challenge. This is where our research comes into, our means not mine, the entire universe, believe me, comes into picture, handy. And a couple more challenges I'm just narrating here: the polarity of the solvents that we use, sample matrix, and then how the molecules are interacting with the other components in the system, and then what is the temperature you're using, what is the pressure you're using, techniques, particle size, whatnot. All these things plays a major role. And also, as I said earlier, the amount of these compounds is quite low. For example, if you look at the potato research that I showed you, the 2014 paper, the amount of polyphenols present are only around 40 milligrams. It take 200 grams of a potato, it contains around 50 milligrams, roughly around 100 milligrams of this polyphenol. That does the required trick, but for us to extract them and then use them for a large scale applications, it would be quite challenging. Okay, and also one more thing, always don't be under impression that we have only one variety in a given system. It never happens. We'll have a distribution. So if you want to separate them out, once again, so the research is completely, there are new techniques have been developed, but still some more development is needed. And then also, most importantly, the type of extraction that we are adapting. We are using supercritical failure, ultrasonic, or micro extraction, pressure, solvents, whatnot. So for every, don't be under impression that we have one unique solution. Not have. We may have to use combination of all these techniques to extract these compounds. So now let me get into my research, if you don't mind. I mean, I hope that you all got some background about these bioactive molecules, their marketability, and then the interest why it had to do. So initially, we were focusing on anthocyanins from Aronia berries. The Aronia berries predominantly they are grown in the US and Canada in the cold regions, and predominantly there are 3 varieties. The interesting thing here about is the plant could survive for about 15 to 20 years. The lifespan is that much, and then they could. Each plant could give anywhere 15 to 20 pounds. It grows only around 6 feet. That's the interesting thing. And if you look at it's a purple color. The most interesting thing is it's called as a superfood mainly because, it's called as a superfood mainly because it is rich in anthocyanins. It is at least 4 times more that of blackberry. And then a wide varieties of food products that could be jams, jellies, wines, teas, whatnot. Many of them could be individual. And these are a couple of papers. I mean, there are many papers that highlight their health benefits like antioxidant properties and other things. So as I said here, this table, it clearly shows that compared to blackberry, there are around 4 times higher, and then compared to strawberry, I think 18 to 20 times higher anthocyanins. So which is really good. But however, the challenge here is why is it not fine in the market? Why? The reason is, it is the moment you bite it, you get a pungent, the astringent flavor. It's too strong that we'll try to spit out the berry. That is the challenge. So that is where we need to mask those anthocyanins, the flavor, so that we could use them in different food applications. But nonetheless, now we have some good amount of wine and then juices. But during that process, believe me, all the amount of anthocyanins present in a berry are not being used. During the preparation process, we are losing a lot, but still it is better than the other products. So here, what we try to look at it is, we wanted to understand what is the effect of location and growing conditions, and also amount of harvest, how we could have whether it has any effect on the total amount of anthocyanins present in the system. So the interesting thing we found out was that as a function of harvesting time from July to September, in 2 months, normally people used to harvest. So late harvesting is always better in terms of anthocyanins. Okay, I'm not talking about Brix value. I'm not talking about the fruit size and all those things. Just we focus here only on the amount of anthocyanins, but one has to do an optimal study of the size and other factors when you consider. But in terms of anthocyanins, it clearly tells you late harvesting is highly effective. And furthermore, the interesting thing here is, as expected, the location has a significant role. I will not get into those details. If anyone of you are interested, we could discuss it later. But it clearly tells us the soil conditions, the temperature it is reading, the sunlight it is reaching, and we have used same amount of fertilizer. So in terms of fertilizers, there is no difference, but the soil condition plays a major role, and then moisture plays a major role, and some of them have rainy season also in those regions, and consequently, it looks like all that have affected the amount of anthocyanins present. And now our goal is, I know already in the market there are some capsules based on this, because they're just drying the berries, crushing them, and putting them in the capsule, and then people are taking it. I mean, that is in the market. But however, we are currently focusing on developing breads based on this. And then I don't have the results to share with you. In the interest of time, I will move on to a different concept. And then we also working on DDGs, dry distilled grains, because when we use corn in the ethanol industry, all the starch is being utilized for the ethanol production. What we left in the grain is a protein-rich compound. Along with the protein, there will be some bioactive molecules as well. So my colleague, Dr. Krishnan, is interested in using that protein in developing a food product, which I also have a little interest, but I'm more interested in this bioactive molecules. So because during this washing process, we would like to take out any toxins and all those things, but those bioactive molecules also will come out when you use different solvents. So I'm trying to reconstitute them and then recapture them and then try to develop the food product. So we made a lot of foods based on this. For example, we made we made cookies, and also we made chapatis and naan based on this. They are rich in protein, because the DDGs, if you look at it, there are around 46%, 40 to 50% of protein is present in them. So we could nicely exploit them, but however, we cannot dump a lot of protein in the system, but we could add, and then taste-wise, we did the sensory and everything. There is no difference at all. And then people love it. And we even looked at, prepared the breads, and then we even looked at the glycemic response and all those things. This is where my research lies in preparing the breads. So when I talk about the bread, we even focused on using the product development point of view, functional food point of view. We prepared a sweet potato-based bread. Why sweet potato? As we all know, sweet potatoes are rich in beta-carotene, and then that helps improve the eyesight. And in South Africa, in some parts, and my collaborators in Nairobi, Kenya, and then there, that region is fighting for a long time with the eyesight issues. So to address that, they have developed these sweet potatoes, orange sweet potatoes that are rich in beta carotene. So we took that, and then when this Daniel, he visited my group, we substituted the wheat flour with this sweet potato puree, and then we made the bread. We could substitute up to 50%, above 60%, 50%, we could not make bread because bread loses its structure. So we got a very nice structure, and we could even understand how much of that beta carotene is available after digestion. But most interesting thing for me out of this exercise is the starch digestion, because you see, this is the amount of starch digestion as a function of time. And then as you add more and more amount of sweet potato puree, the whole starch digestion reduces. That tells us that sweet potatoes, though they are potatoes, the sweet potato consumption is really good for health. And then you all know the starch nutritional profile. If you look at it, we could categorize them based on the rapidly digestible starch amount, slowly digestible starch amount, and resistant starch amount. And in here, we found out that rapidly digestible starch amount reduces significantly, and then this resistant starch, which is a fiber, that increases significantly, but there is not much effect on the slowly digestible starch. So that tells us that when somebody consumes this bread, there will not be a surge in their blood, the amount of sugar, which is really interesting. And also, it contributes to fiber. So this will be really healthy. So this is the very good outcome of the study, because now this sweet potato consumption, the bread, will not only help to improve the eye problems, but most importantly, it could even address the diabetic issues. The reason is mainly because the amount of beta carotene present in the system. You remember a couple of slides back, I told you the potatoes with variety of this the molecules, nutraceutical compounds, that the more amount, less starch digestion. Same thing is happening here too. So the sweet potato puree has more amount of beta carotene. So the more amount of beta carotene, that neutralizes the starch enzyme activity, and thus the whole starch digestion reduces significantly. So this, with this idea, now we are growing sweet potatoes in Uganda now. We identified around 8 to 10 cultivars. We are growing sweet potatoes, and that puree we are getting, and then we are making breads, and then ultimately these are, by the way, these are all in vitro studies. And then these in vitro studies, once we identify a system that could reduce significantly the starch digestion, then we will be doing the human studies and all those things down the lane. And now moving on to bioactive molecules, the carriers, because when we want to develop the functional foods, we cannot just like that add the food system, because at least my previous sweet potato research could have told you that, could have convinced you that the bioactive molecule bioavailability during the digestion process changes significantly, and that's where we need to have the carriers. And there are wide variety of carriers that people have been using throughout the world, like spray drying, conservations, liposomes, inclusion compounds, crystallization, micro and nanoparticles, emulsions, hydrogels, variety of systems depending on the need. The whole goal, if you look at it, as you all understand, the structural form of the matrix, the bioactive molecule, that needs to be preserved, and then it has to be delivered at the active site effectively. That is the whole goal here. So a couple of examples, if you see, in the case of nutraceuticals or diterpenols, the good examples that I could think of are protein-based hydrogels, but there are other systems also. But for the discussion point of view, I chose protein-based hydrogels. So these are used heavily. These hydrogels are being used, but unfortunately, hydrogels don't have a stable structure, as we all know. And one more thing, we at this moment, the knowledge about protein-protein interactions and protein-polyphenol interactions, we don't have. So that really lacks, that impedes the overall design and development of the diphenolic based carriers. And in the case of vitamins also, we have a lot of nanoparticles and as a carriers, and predominantly all these nanoparticles are coming from the non-GRAS materials, generally recognized as safe materials, so which is not good. If you could develop a starch-based nanoparticles and use as a vitamin deliveries, that would be awesome. But I think there are very few examples are there. And some of you might even work on, like to listen about the drugs. And then we know there is a lot of research on synthetic molecules and biodegradable molecules. But however, we are aware of the side effects, and then there's toxicity associated with this, and then also high amount of cost. And this is where, if you could come up with a system that is human compatible, and it also could maintain the structure, that would be advantageous. So this is what carriers with stable architecture and general advantages to retain the structural form are in need. And that is where I sincerely believe carbohydrates are one of the alternatives. Though you might say I'm working in carbohydrates, so I'm biased, but I will convince you that carbohydrate, because we have been consuming for carbohydrate for generations, nothing has happened. So these are all GRAS materials. So if you could use this GRAS material as a carrier, that would be advantageous. That's what I would like to say. So we know the carbohydrates, carbohydrate means don't mention only starch. Non-starch polysaccharides could be categorized under carbohydrates. We have been using them we could get them from nuts, seeds, the extruded gum that you all might have used this gum also just to glue the papers. That gum is rich in protein, to be frank with you. And then that arabinoxaline also it contains, and that would be a good source of, people even use some areas to as a food. And many of you might be aware of this shrimp. And the shrimp shells, if you look at it, that contains the polysaccharide called as a chitin and chitosan. That is the only positively charged polysaccharide that we know as of now. Otherwise, all other polysaccharides are neutral or anionic polysaccharides. So that is also used civil. And then seeds, and then leaves, whatnot. Simpson is having a lot of burgers here. The starch we use a lot. So all these things we get from varieties of sources, and we have been using them in our food application. I think many of you like ice cream. Ice creams, we use a lot of this food application, part of this polysaccharides, carrageenan, xanthan, or anything. And then even skim milk, and then emulsifiers, we use them as emulsifiers. And then we are also using as the protective casings, and also as the capsule casings. And then the beer also contains glucan and whatnot. So all these things people have been using for a long time. So if we could use this, these are generally recognized as polysaccharides, so human compatible too. So if you could use these human compatible molecules, biopolymers, as the carriers of this health promoting and disease preventing compounds like these nutraceuticals, that would be advantageous. But challenge here is these compounds, these polysaccharides, they don't possess organized structure. They're also like flexible molecules. But interestingly, we could create an organized network. That is my research. So if you look at the chemical structures, the cellulose and amylose, cellulose is nothing but that every plant material that we see the cellulose material, and amylose is a starch that you and I digest. And then if you look at the chemical structure, the difference is, one is, in the case of cellulose, the linkage is the hydroxyl groups in the equatorial, that is gives you beta. When it changes to axial, that gives you alpha. And then our human system, it could digest, could hydrolyze these alpha bonds, and consequently we are able to digest that and then get the energy. And then if you use any cellulosic material, beta cellulose, our alpha amylase cannot hydrolyze, and then that goes as a fiber. And on the other hand, animals could consume, could digest both. Predominantly, they digest this cellulosic material. So, but still, by looking at the chemical structure, we cannot really understand their properties. But that's where one has to solve the structure and understand the properties. And then the structure-function relationships is a completely different domain. I think that is not focus of this talk, so that's why I cannot talk. But otherwise, predominantly my research focuses on solving these three-dimensional structures and understanding the functionalities. But now for today's talk, what I'm saying is, using these polysaccharide matrices, we can create organized networks. And then when you create that organized network, during that process, there will be some pockets, water pockets created in the network. They're not random, they're highly organized. Those water pockets that is predominantly rich in amorphous water, that could be nicely encased with the bioactive molecule, and then that could be used. That is a hypothesis of my compound, my research. So here, for example, these polysaccharides, as I said, they don't have any organization initially. It is like this. One example I can think of is, let us say in a conference room or in the classroom, all the students are entering at once. But however, after some point of time, when they sit nicely in the seats, there is an organization. So so if you could call this as amorphous state, this is a highly crystalline state. Okay. So these molecules could be tweaked to get this organized network structure. So during that process, depending on the molecule we are working, we could have single helices, double helices, triple helices, and then the chains could be connected through cations, a series of water molecules. That all depends upon the molecule that you are working with, that is the polysaccharide that you are working with. For example, here this is iota carrying, this forms a double helical structure, and then chains could be nicely connected through a cation like calcium ion or sodium ions and all those things. And then you focus on these empty spaces. These empty spaces, that's where the amorphous water is present. There is no crystalline water. So that pocket space, if you look at it, anywhere between 25 to 50 angstrom cube. Okay. So this is a three-dimensional space. So that nicely, it could encase a bioactive molecule. And then if you could create these complexes, the molecule once stays in the network, that gets all the protection. So thus, these carriers, if you use them during the storage process or product development, that would help us to protect the molecule from the external stresses. That is my focus of my research. And for a long time, I have been fascinated by curcumin and then resveratrol because of their health benefits. And predominantly, all my research focuses on these two only. We are using different matrices, but predominantly we do this. So initially what we try to do is we try to use the curcumin. We encapsulated them in the sodium iota carrying fibers. I'm saying sodium mainly because iota carrying is an anionic polysaccharide, and then we need a charge valence cation. We use sodium ions. We also could use potassium ions, calcium ions, but that research is a little different, which I'll show you. And then I'm pretty sure many of you would have seen that fiber diffraction pattern. I think the famous DNA diffraction pattern, where the Watson, Crick, and all of them, Wilkinson, they got the Nobel Prize in 1953 for their DNA structure. And same way here also, I was fortunate to be part of that group, because the Wilkinson, the King's College London, that's where the Strother Arnott, he worked. And then he came to Purdue University, and then with him, my boss, Dr. Chandra, he worked, and then they started, they ventured into polysaccharides, and that's where I joined. And then it's a long story, but we are all crystallographers. That's what I can tell you. So this diffraction pattern, if you look at it, these are all the, these spots are called as reflections, and they're all arranged in the layer lines, if you could see it. So what we did is we encapsulated curcumin, and then we also wanted to understand the effect of temperature. So we did it for the temperature, the complexation. You just focus on the second layer line. So this is where the reflections are very well resolved. That clearly tells us this organization, the structure is very well resolved here compared to other systems here. But as I said, the details I could skip if you don't mind, because if anyone of you are interested, we could discuss. And the third layer line here, this reflection that is called as meridian reflection, the third layer line is same. So that tells us that molecular structure hasn't changed, but only thing is the molecule has entered the spaces, and that has altered the what you call as basal net dimensions. That's what really happens, because when the molecule enters the space, There could be some small alterations, but the shape hasn't changed. So we later looked at the release profiles in the aqueous conditions. We could clearly see that complex at 25 degrees centigrade encompasses more amount of curcumin compared to other things. But all these differences also could be explained based on the structural arrangement. But most interesting thing came out from the melting. So curcumin melts at 170 degrees centigrade, but however, when you put it in, when you add encapsulate in the fiber matrix, we don't see any melting, but instead there is a crystallization, mainly because that is amorphous state, and then thus during heating, and then it looks like it reassociates with the carrageenan chain, and then it gives you a new system. So this tells us that once the molecule is in the system, that gets protected from the heat. This is very interesting thing, and then this was published in 2012, and then there were a lot of comments, especially the first news release that was written by some researchers, just smuggling medicines in food, because any compound of interest could be added to the system and then delivered, which is really true. And we even showed that later that we have to have drug molecules and hormones, anything could be added to this sodium iodide carrying fiber and then create fibers and network structures and then use them to deliver. So you might be thinking, oh my goodness, this is a very good, very interesting research. But there is a small caveat here, to be frank with you. The amount is very low, very, very low. It is only micrograms per milligrams. This is quite low. So technology works, but the practicality, it fails. And this is where we were working how we could increase the load. I'll show you an example. So this was, this was recently, we just published this. No, later in 2022, we also worked on the, we wanted to understand what is the amount of time required to encapsulate the maximum amount in the system. So once again, we worked on the curcumin and resveratrol. For about 5 weeks, we encapsulated, and I'm showing the diffraction patterns here. And then this is for the resveratrol. And then we looked at the release profiles. I mean, I'll skip all those details. We looked at the release profiles, once again, in the aqueous conditions only. And then all these things could be nicely modeled, the release kinetics and all those things could be analyzed. But by far, it looks like we could explain all those things based on this first order release or Casper model. The first order release is nothing but diffusion mechanism. So you have a matrix and then a molecule comes out, which makes sense mainly because this iota-carrageenan matrix we have, and then resveratrol molecule or curcumin molecule, it is nicely slowly coming out. But there is a different stage, 2 stages of releases are happening, by the way. So in the second stage, what really happens is after swelling, the molecule, we are also losing the network. Even that makes sense mainly because we have used a water-soluble iota-carrageenan matrix, and thus that after some point of time, it loses its network. This is really interesting mainly because the interaction between the curcumin or resveratrol molecule with the carrageenan matrix really dictates how long we could hold that matrix for a long time. So this will have a lot of implications, we believe, and then based on this, now one could develop a lot of emulsions or any beverages or any food products. That's what is our next focus. And also next, we tried to ask ourselves, okay, a question. Now when the molecule is encased in this matrix, there is a charge. For example, here calcium ions. Instead of calcium ions, if you have potassium ion or sodium ion, how does that affect the total amount of encapsulation? So what we did is we used both sodium and calcium ions, and then we try to, but instead of curcumin and resveratrol, we used eugenol. Okay, we are using wide varieties of molecules just to expand the research. And then in the case, you see now, the total amount has increased up to 3.5 micrograms. And then but still the maximum release is happening within 30 minutes. But on the other hand, when you have calcium form, for a certain point of time, there is no release. Molecule stays put, and then there is a release. We haven't analyzed the release kinetics to show you at this moment, but it could have convinced you that both networks are different. So that could be explained once again based on this network formation and all those things. Okay. The most interesting thing here is other divalent ions like zinc, calcium, strontium, magnesium, if you use, especially in the case of nickel, we could encase up to 140 micrograms of carvocol. The carvocol and eugenol are the essential oils that have antimicrobial properties. But so far I have showed with you 4 results: curcumin, resveratrol, eugenol, and then corocol. So four different properties, and it looks like the corocol could be encased more. So it all boils down to the interactions that are taking place at the atomic level between this bioactive molecule and the curcumin towards the product development. Always remember that. And then further, what we tried is we further wanted to increase the space in the carrageenan matrix. So that's where we try to use this cyclodextrins, alpha cyclodextrin, beta cyclodextrin, gamma cyclodextrin, and then see how that impacts the overall load. Especially in the case of curcumin, we found a significant increase. Tomorrow, my student will show these results in detail. But what I want to tell you is, by playing with the network, which it looks like we could increase the loading of these bioactive molecules that could be stable. And then based on this, one can develop the food products. That is our next focus. And then apart from this polysaccharides, one can also use starch, because starch you and I eat on a daily basis, because everything that we think of is based on starch only. So if you could use the starch as a matrix, and also, the consumption of starch is the one that answers the diabetes. So my goal is, you eat the starch, but at the same time, you add some bioactive molecules to it so that it reduces the overall starch digestion. So I am using starch as a vehicle to deliver this bioactive molecule. That is a novelty of this research. So and as we all know, starch predominantly we get from the cereals and then tubers. For example, cereals in the sense of rice, corn, wheat, that they grow above the ground. They're all called as cereal starches. So they have one type of network. On the other hand, that go below the ground, like tapioca, potato, or sweet potato, they'll have a different type of network. You see, this is a starch double helix. In the projection, I used it just as a circle, and then this black dot is a water molecule. So in the case of cereal starches, we have very tight network arrangement. On the other hand, when you go to these root starches, we have the water channels around that is around 16 to 17 angstroms water channels. That is a property of the root channels. Starches. So these root water channels could be used to encase a bioactive molecule. That is the focus of my research. So what I did was I used this potato starch, cassava starch, that is also root starch only, but this predominantly is C starch, and this is high amylose maize starch. This is also B starch, but this acts as a cereal starch because now it is above the ground. And then waxy maize is nothing but a corn starch. Okay, so we use this, and then we encased this both curcumin and resveratrol, and then as expected in the case of potato only, we see significant reduction. Cassava, we see some starch digestion. In high amylose maize and waxy maize, because of the cereal type networks, we could not encase and then get any. So this is, see here, in the case of potato complex, the overall rapidly digested starch amount has significantly reduced, and then SDS hasn't changed much. But RSCC, there is a general resistant starch that works as a fiber. So when you encapsulate a bioactive molecule in the starch matrices, we get a lot of fiber increase. Same thing you would have noticed in the case of sweet potato bread also. As we increase more amount of sweet potato puree, we are seeing more amount of resistant starch. And then slowly digested starch also has reduced, same thing is happened. So it looks like in the matrix, in the starch matrix, if we have this bioactive molecule, the rapidly digested starch amount significantly reduces, and whereas resistant starches increases, but there will not be much effect on the SDS. That is a very interesting observation. And now that we are using to make products like breads and all those things. But another research I would like to share with you is this potato starch, porous starch based carriers. Because you see in the B network, if you remember, I said there are around 17 angstroms water channels, and those channels that could limit the molecular size, because 17 angstrom. If it is more than that, it cannot go. So that is where what we are trying to do is we are taking the starches, corn starch, and then we are enzymatically modifying. We are creating some pores on them. So you see, they see nicely pores. The pore size could be anywhere from 1 nanometers to 10 microns or 20 microns. So this is the corn brands, the corn starch. We could use rice, wheat, or anything. That is. So once you create these pores, what we could do is if starch granule. If you could imagine like a tennis ball. So tennis ball inside there is a lot of space. So here, when you create those pores, now all the inside space is accessible to us. So this is what we are using to encapsulate a bioactive molecule and use this porous starches and add them to food matrix. Once again, we're focusing on the breads only. So we try to use the turmeric here, and then we added them to bread matrix, and then the turmeric, the amount of it's released from the matrix is shown here, and then most importantly, the starch digestion in the presence of turmeric, overall starch digestion reduces. So this is another way of developing food products. So we are using starch as a carrier matrix to reduce the starch digestion. That is very interesting, but some people will not like it, mainly because why are you using starch as a carrier matrix? Because we want to address the starch starch properties. We want to modulate the starch digestion, but all of a sudden you are using starch itself to modulate its digestion. So it's hard to envision, but this is this really gives us a good significant results, and especially to address the diabetic issues. That's where I'm heading to, because I'm very much interested in addressing these diabetic issues if possible through these foods. And we all know that by 2025, I think in 10 more years, now 22, and yeah, 13 more years, at least this planet will be filled with 500 million diabetic patients. That's what is projected. And there could be undiagnosed patients more than 1 billion, I believe. And in 2014, world has spent around $612 billion US dollars to address this diabetic concern. So if we could use that amount for a lot of research scholars, we could have a lot of faculty, we have a lot of facilities one can think of developing it. So that's where I believe once we develop these products based on these bioactive molecules, that could help. The diabetes is the one issue. I mean, there could be a plethora of other health diseases one can think of doing. So overall, my research, if you look at it, I am trying to create the organized networks based on the carbohydrate matrices. It could be starch or non-starch polysaccharides. And though we are predominantly focusing on the dietary phenolics, and then also we did some drug molecules, I'm also interested in the antimicrobials and flavors and vitamins to encapsulate them. And then later we'll use those, we are using those complexes to develop some novel foods. Most of my research has been on the breads. Some of the bread examples I have shown you, but other things also we are focusing. But I'm also dreaming, this is my dream, to be frank with you. I would like to have this encapsulated, this bioactive molecules matrices in the powder form, and next to salt and the pepper, I would like to put them, and then whenever we want, we could just like that add them to our food matrix like salt or pepper, and then consume. That's my dream, and hopefully we'll be able to do it. I think that's all I have. Maybe I thank all this. And as I said, these are all my students and my collaborators. It's not done by myself. And then I thank this Borlaug Fellowship for the sweet potato research, and then SDSU and then NIFA for this grant. And then all the X-ray diffraction patterns were collected at the Argonne National Laboratory, Chicago. And then that's all I have. And then I'll be happy to take your questions and then comments. Thank you.
Thank you, sir. Excellently explained about the bioactive compounds as suitable foods towards developing novel functional foods. Now the session is open for questions or any queries or doubts. You may post it in the chat or directly you may ask.
If you don't have questions, I'll ask.
Sure,
Based
sir.
on my research are general things. So please come up with some questions.
If you have any problem, post it in the chat, please. In the YouTube link also, you may post the queries.
That's fine.
Arna madam.
Hello.
Yes.
Sir, I have a question, sir. Sir, have you carried out with Gurgaon and also the tapioca flour? Because it has been assumed that this junk foods, whatever the people are consuming, it is containing more large quantities of tapioca.
The tapioca starch, yeah, as you could see, are you seeing my screen here, the cassava starch?
Yes,
There's nothing
sir.
but the tapioca starch. But the variety that we got, this we got from Ghana. And as you know, there are several varieties, and then depending on the cultivar we have, it could have different network arrangements. So the one that we got is predominantly close to A starch. That's why it did not encapsulate much. But as you rightly pointed out, it could be made as healthy. I'll tell you, but though it's used as a junk food at this moment, it could be made as healthy by complex with more compounds, bioactive molecules. So we did with one cultivar, but we need other cultivars. We had to study with other cultivars, which we could not do at this moment.
Okay. Sir, what about these gums? You have already mentioned that some of the
gums predominantly, as I said, I have been using this carrageenan.
Carrageenan, yes.
I have been using this the carrageenan that comes predominantly as seaweed polysaccharide.
Yes.
But we could use other systems also. But that depends on the network creatability.
What about this cluster beans gums that is available with the cluster beans? That is locust bean gums.
Locust bean gum. So locust bean gum, I have not focused much, but one can do that because locust bean gum.
Because usually in India it is available in plenty, sir. If it is being used for diabetic patients, I think
locust bean gum predominantly it forms a sheet-like structure, and then, but still we could create, depending on the variety that we are getting, there will be some pockets available in the network system that could be used to encase a bioactive molecule. One can do that.
Okay.
But I have not, but only thing is I have not focused because majority of my research was on the carrageenan. But locust bean gum is also a very good example that one can use.
Yes. Very good information you have given, sir. Very nice information you have given.
Oh, thank you.
For a researcher, it will be very much.
But that also depends on the variety that you are getting in a particular year.
Yes.
Mainly because there is a year-to-year variability we have seen in the case of locust bean gum as well, and then that might change the network structure. It will not change the network structure, but amount of water pockets that are available to us to encase could change.
Okay.
But still, that is also worthy to investigate.
Yes.
Yeah. Sir, one question is in the chart. That's.
Can we use the same technology for rice as it is a cereal starch and is staple in our country? Thank you. In the case of starch, the Starch Network are you able to see the screen? The starch will have a A-type network, and thus the pockets are not available for us to encapsulate. But however, we could nicely create these porous starches because rice granules are around 10 to 30 microns, but still we could create some nanopores on the rice granules, and we could encapsulate. We could, once we create those pores, we could nicely add a bioactive molecule and we could use it. To say, if you want to say yes or no, yes.
Sir, how much percentage of sweet potato you have added into the bread, sir?
We used around up to 50%.
50%. It came out very well, sir.
Oh,
with
yeah.
the porous structure and all.
Yeah. See here the bread.
I have seen it, sir, that figure. But regarding the taste, sir, it
is
the taste
very
parameters.
Good.
Yes. Okay.
Even the color is also more appealing, sir.
Yellow color, yes.
Yeah. Sir, one simple doubt, sir. Generally, even in our university, in Rural Women Technology Park, we developed some products with tulsi leaves. What should be the best time to collect the leaves, fresh leaves, sir, from the plant to extract the bioactive compounds?
I did not work on them, so I don't.
Yeah, it's generally I'm asking, sir, whether you have any idea.
Oh my goodness. I'm sorry if I say something, it might be wrong. So let me not.
It's okay, sir.
But I don't know. Sorry. Because I haven't worked on that, so I don't want to give you any wrong information. But other than if it is structural point of view, I can say, but I have not used that. Sorry.
Okay, sir. Okay, it's okay. Thank you.
This sweet potato bread, whenever we prepare, whole department, whole building will be in our lab. They want to consume this because it is so the flavor is so attractive. People like it. So it would be really hard for us to save the sample from others because they'll come in and finish it. It is so, but now we are looking at the textural properties and all this.
Here that papaya is always available with us, sir. So if it is possible, we can also prepare the proportion, whatever you have mentioned for papaya bread. We are planning out that.
Sure, papaya is also a good source papaya of this
pulp, yes, beta carotene, yes.
Yes, beta carotene is there, so that would be awesome. Papaya,
yes. Because it also has many health benefits, sir. That is what we are planning.
Sure, do that. That will be awesome. Yes. And I sincerely believe you'll have a lot in terms of starch digestion. There will be a significant reduction.
Yes.
So that would be a good contribution.
Yeah.
Yes.
That's worth pursuing.
We are planning out for incubation center, sir. In that we want to prepare this papaya bread and also the cookies, cakes like that with papaya.
That's really good.
Very interesting research outputs, sir. Yes, we are happy to know about your research.
Yeah, but instead of focusing on one concept, I gave you different examples. Hope you all.
Yes.
The idea behind that is just to give you all a perspective what one can do. So that's what we are focusing on, different concepts, but the functional foods based on the carriers. Hopefully, in about the next 5 years, we'll have those answers.
Yeah. My researcher, she has developed yogurt with different fruits and also carrot, beetroot, yogurt, and also the sapota. So all these yogurts were developed in a very good manner.
That's good. Okay.
Probiotic yogurts she developed along with
do you do any human
yogurt.
trials, human feeding
yes,
as well?
Yes, we did it.
That's
and
good.
It came out that the cholesterol levels have been gone down when we use these high-risk patients, cardiovascular disease conditions.
Congratulations. Okay.
Thank
you. Shall we conclude, ma'am?
Yes.
Thank you, sir.
Thank you.
You elaboratedly explained about this mainly functional foods and nutraceuticals, and mainly you well said about healthy compounds are mainly present in the outer layer of the food, that is particularly in the fruits and vegetables. So we have to consume that one. And
Sirisha, there is a question in the chat box.
Yeah, one more question in the chat box.
Okay. Yes, sir. One question.
Yeah, pumpkin puree. Yeah, you could definitely use pumpkin puree because that's also good in beta carotene. The pumpkin and then papaya, they are very good. Pumpkin is really good.
Yes, yeah.
I'm not really sure whether there are any breads in the market, but pumpkin will be really good. Yeah.
Yes, dear participants and delegates, if you have any questions or queries, please ask. Now we are concluding the session. Yes. So mainly specific compounds and its functions, challenges in extracting the bioactive compounds. You said very well, sir. Extraction methods also. So we have to use combination of the extractive methods for the collecting of these bioactive compounds. And Aronia berries and anthocyanin compounds and its importance, and food grade DDG and its food products, sweet potato breads, carbohydrates are worthy alternatives. Yes, sir, I also agree with you. Curcumin encapsulation, all these things you explained very well. Now I invite Dr. Rajani Madam to conclude the session and present the concluding remarks and vote of thanks. Welcome you, ma'am.
Thank you, Sirisha. Yeah, it's a pleasure to be here to give the concluding remarks of the inaugural session. First of all, I would like to appreciate and thank the convener of the conference, Dr. Sirisha, and the other team members for choosing very relevant theme on nutraceuticals and functional foods for human health. So we know that nowadays the people are very much conscious about their health, and paramount significance has been given for the healthy food and for the health also. And today all our speakers explained in detail about the nutraceuticals and functional foods, their role in the human health. Yes, nutraceuticals being an integral part of component of functional foods and a physiological arm of contemporary medical treatment, it's a challenging frontier in science. That's what our keynote speaker also mentioned as an emerging scientific community research that calls for a strong union of nutrition and pharmaceutical industry, wherein the efficacious binding of prebiotic, probiotics, and particularly dietary fiber, alongside of the what we call dairy products, could be an answer to the treatment of the chronic diseases and in recuperation too. Okay. And even improved health, delayed aging, increased life expectancy, as also as a curative elements in allergies, and cancer, diabetes, Alzheimer's. In all these diseases, nutraceuticals are creating a renewed confidence in the scientific community that new avenues of treatment are there. Functional foods and nutraceuticals demand also increased tremendously during the COVID-19 period also. It is very clearly visible for all of us also. There is a shift even in the food consumption pattern also, because the people are preferring now the healthier foods rather than only to satisfy their hunger. So that shift was really observed during the pandemic time. In fact, the nutraceuticals are being regulated as drugs for use in several countries, even including America. The herbal components of nutraceuticals has a strong base in India. The patent angle has to be, only the patent angle has to be firmed up and regulated for intellectual property rights also. And I think all of us know nowadays the nutraceutical industry in India started showing little positive results also. Functional foods and nutraceuticals are developed through Sir also, our keynote speaker, rightly mentioned that different methods, novel methods, that means I can say that the design thinking methods are necessary, because those methods will focus on the human health problems instead of focusing on the technology or organization. So definitely the researchers, academia, and the food industry that will develop a new functional food and nutraceutical products, it is very important to plan and know the targeted consumers. So suppose if we know the targeted our consumers, so that the product also will be successful and it will reach the consumers and helpful for the concerned consumers also. And in today's inaugural session, even our head of the department, Professor Aruna, has mentioned very clearly about the activities of the department and what type of activities we are doing. And we are doing a lot of research work, and we are trying to reach the community also through our products. Definitely we'll succeed in our efforts. And our registrar, Professor D.M. Mamata Garu, she also rightly mentioned that what you eat, tell what you are. Yes, it's true, madam. The food, what food we eat affects our health, energy levels, mood, and behavior also. And madam has explained clearly about the novel products development for the human health, their importance, their significance also. Then the Dean School of Sciences, Professor Nagaraj Garu, he presented very relevant to the present day situation. And as a pharmacy expert, sir has made very much very good expert comments on nutraceutical and functional foods, and mentioned about the government programs that could address the hunger, and about the market scenario, marketing the market competition for these products also. And finally, I would like to mention here again about our keynote speaker, Dr. Srinivasa Janaswamy Garu, for highlighting about the bioactive compounds as a sustainable tools for developing the novel functional foods. Sir, really, you gave a different perspective and more insights into the bioactive compounds, nutraceuticals also. Thanks a lot for your enlightened and effective presentation regarding bioactive compounds and the consumers' demand also. As you said, it is the day of consumer demands. Present days, the consumers are demanding more safe and healthy foods. And a lot of research is also going on to evaluate the effects of bioactive compounds on health. So as a keynote speaker said, bioactive compounds have a significant role in preventing and treating the diseases also. Definitely this information will be more useful for all the participants. So really, I'm very happy to conclude this inaugural session. Now it's the time for a vote of thanks also. It is indeed a immense, it is a matter of immense gratification and honor to extend our sincere thanks and regards to our Honorable Vice Chancellor, Professor Jamuna Duvuru, for her close support, close cooperation, support, and encouragement given to the Department of Home Science for conducting a very useful international conference. And we sincerely thank you, Madam. And we sincerely recognize the support provided by our registrar, Professor D.M. Mamata Madam, for conducting the seminar and for her special message and extending her views on the nutraceuticals and human health also. And I acknowledge the support provided by our Dean School of Sciences, Professor Nagaraj Garu, and for his presence on this platform and his valuable words. And my sincere thanks to Dr. Srinivasa Janaswamy Garu for enlightening us on various aspects of bioactive compounds. Sir, really having you on this platform made a huge difference and provided a right perspective related to the bioactive compounds. Thank you very much, sir, for accepting our invitation and for your presence here. And my special thanks to Professor A. Jyoti. She is a former Dean School of Sciences and a former head of the Department of Home Science in Sri Padmavati Mahila Viswavidyalaya. Madam, thank you very much for your presence on this platform. Madam is always very great support to the department, and she's a well-wisher for the department also. She contributed a lot for the growth of the department. Thank you very much, ma'am, for your presence. And I convey my profuse thanks to all the speakers who have readily accepted to give their to deliver their talks. Dr. Pushpa Bharati Garu, Naveen Kumar Rastogi Garu, Dr. Shushil Kumar Sharma, Dr. Manjula Kola, Dr. Hulu Jo, University of Massachusetts. So without cooperation of the resource persons, it's not possible to conduct the conference very successfully. So once again, I thank all the resource persons for accepting our request, and my appreciations to the convener, Dr. Sirisha, and the team members, and for all the department staff also for putting efforts for organizing such a very apt conference, and for the sincere involvement also. And finally, all of us know thus that all this would have been impossible without the participation of the delegates. So I thank and appreciate for the sincere involvement of all the participants. So Sirisha, Dr. Sirisha, once again, I thank you for giving me this opportunity to conclude and propose the vote of thanks also. Thank you.
Thank you, ma'am. Thank you so much for your cooperation. Thank you, sir. I think it's midnight in your place. Thank you for your presence.
No problem.
Thank you, HOD ma'am, and thank you, Jyoti ma'am, and thank you all the participants. Now we will break for tea, virtual tea. So 10 minutes we are giving for break. So after 10 minutes, dear participants, all of you must join for the second session. Thank
Yes,
you.
sir. Dr. Srinivas, sir, we look forward for your cooperation, sir, in future
Oh,
also
sure.
for the department.
Sure, I will be very happy. I will
Yeah.
be very happy.
Thank you.
I have already included my email, so feel free
Yes, sir.
to contact me at any time. And then I once again thank you all for giving me the opportunity. And then it's a pleasure seeing you all.
Thank you, sir.
Thank you,
Thank you, sir.
and good day. And
Thank you,
then
sir.
wish you all success in successfully completing this, conducting this conference, and then all the best.
Thank you.
Thank you,
Thank
sir.
you.
Thank you so much.
I'll take leave now.
Yes, sir. Thank you, sir.
M. Sirisha, congratulations.
Thank you, ma'am. We are
Congrats,
very
Sirisha.
support. Thank you, ma'am.
Congrats.
Congrats. Okay.
Jyoti madam,
10 minutes break.
Jyoti madam, thank you, Jyoti ma'am.
Jyoti madam, unnara ikkada?
ఉన్నారు
Ah.
ఇంతసేపు ఉన్నారు.
Okay, Sirisha. Thank you.
ఓకే శిరీష.
Thank you, ma'am.
జ్యోతి మేడం ఉన్నారు. జ్యోతి
Ah,
మేడం
madam. Mute.
మేడం మ్యూట్ లో ఉన్నారు మేడం మీరు. మేడం
Thank you a lot.
థ్యాంక్ యూ మ్యామ్.
Thank you, ma'am.
Aruna, Rajni,
Yes, ma'am.
Jyoti garu.
recognizing me and inviting me for this.
మిమ్మల్ని రికగ్నైజ్ చేయకుండా
మేడం మీరే కదా స్టార్ట్
మీరే
చేసిన వాళ్ళు
department,
ఇన్సెప్షన్
growth
డిపార్ట్మెంట్
కంతా.
స్టార్ట్ చేసింది
Ah.
మీరే.
అదే.
Thank you. Thank you. All the best.
ఓకే జ్యోతి
ఓకే
గారు.
మ్యామ్. థ్యాంక్ యూ మ్యామ్.
Thank
Oi.
you. Thank you.
థ్యాంక్ యూ.
థ్యాంక్ యూ.
థ్యాంక్ యూ.
Thank you. Thank you.
బాయ్ బాయ్.
Bye. Bye.
Bye.
See you. Flying kiss you.
బాయ్.
Bye.
Bye.
Flying kiss. Flying kiss.
Good afternoon to you all. Ma, message our students to join fast, ma.
Yes, ma'am.
Good afternoon, sir. Now I welcome Beulah to introduce our today's speaker, Naveen Rastogi, sir, from CFTRI, Mysore.
Yes, ma'am, can you share the screen, ma'am?
Yes, ma'am, sharing. Yes, over to you, Beulah.
Okay, ma'am. So welcome all to today's international 2 days virtual conference. So I am so delighted and great honor to introduce and welcome today's speaker, Dr. Naveen Kumar Rastogi, sir. So sir is a chief scientist and head to Department of Food Engineering, CSIR, Mysore, and his field of specialization includes food engineering, food technology, chemical technology, biotechnology, marketing and personal management, maintenance and systems management. And sir interest areas are non-thermal processing, membrane processing, osmotic dehydration, deep fat frying process optimization and modeling, gamma irradiation, bioprocess engineering and product development. So coming to sir's educational qualifications, he did his M.Tech in maintenance and system management with first rank holder and PhD in food engineering. Sir guided several students, includes 10 PhDs, 7 M.Techs, and 10 M.Sc and 25 B.Tech students. Sir received different honors and awards. The list is here. Ma'am, please go to previous slide, ma'am. So he received 2 academic awards, gold medal and merit scholarship in B.Tech in the year 1989, and he also received first rank in university in Master of Business Administration in the year 1991.
Yes, sir.
And he also received 4 professional awards, which includes AFST Young Scientist Award in 2000 and CSIR Young Scientist Award 2001, and he also received AFST Lalji Godanidhi Samarak Award in 2003, and he awarded as in the category of best technology transfer to industry for the production of virgin coconut oil by wet processing. Sir also received 8 fellowship awards, include Seligman APV Fellowship in Food Engineering in 1997 and DAD Fellowship in 1998 to 2000 by German Academic Exchange Service Fellowship sponsored by DAAD, Bonn, Germany. And the other fellows he received Association of Food Scientists and Technologies in India and National Academy of Agriculture Sciences in 2018 and Institution of Engineers in 2018, and the other fellows which includes Indian National and Academy of Engineering nominated by Dr. R. Mukhopadhyay, Director and Chief Executive, Mysore, and National Academy of Science nominated by Professor Alok Dhawan, Director, CSIR, IITR, Lucknow. So, and he also published 170 papers in reputed journals, and among the all journals, the review paper entitled Opportunities and Challenges in High Pressure Processing of Foods, critical reviews in food science and nutrition, which was cited more than 720 times in this data Scopus database. And Naveen Kumar Rastogi sir authored one book by title Recent Developments in High Pressure Processing of Foods, and he also edited 2 books. Currently, he is serving as an editorial board member for 6 journals, which includes Journal of Food Engineering, Journal of Engineering, and Scientific World Journal, Research and Reviews, Journal of Food Science and Technology,
Journal of Food Research and Technology, and Journal of Food Technology and Preservation. He is also an editor and co-editor for different journals, and a CSIR funded FTT project with 300 lakhs was carried out. The topic non-thermal processing of liquid food using high pressure processing, which was carried out for the preservation of sugarcane juice and tender coconut water. And here the high hydrostatic pressure processing facility was indigenously designed and developed at CFTRI with the 1/3 cost of imported unit. Sir has also developed different products like the virgin coconut oil, spray-dried coconut milk, honey-based fruit spread, coconut beverages from tender coconut, coconut spread based on mature coconut, water concentrate, and coconut dietary fiber. Dr. Naveen sir have a significant foreign assignment with different countries like University of Reading, UK to avail Seligam APV Fellowship sponsored by SCI London, UK, and Berlin University of Technology, Germany to avail fellowship sponsored by DAAD Germany, and Montpellier, France at best paper at International Congress on Engineering and Food. And he also have the other significant foreign assignments with DBT India and Aswan Egypt. Sir delivered several lectures on downstream processing of biomolecules, emerging technologies for food processing, recent advances in osmotic drying, engineering aspects of different grinding systems, engineering management with special reference to food processing at various places. So now I request Dr. Naveen Kumar Rastogi sir to deliver his talk on emerging food processing technologies for better retention of nutrients. Once again, I welcome you, sir.
Thank you, ma'am.
Thank you.
Thank you. My slides are clear to all of us?
Yes, sir.
Okay. So we'll be just dealing today about the emerging food processing technologies. And thank you very much, ma'am, for the very nice introduction. And what we are going to deal with about the emerging food processing technologies for better retention of nutrients. And we'll be dealing about the non-thermal processing techniques. And all the heartfelt thanks to the authorities at the Shri Padmavati Mahila Vishwavidyalaya for inviting me. Can you hear? For invitation to present a lecture in the international seminar. And so when we start with this, like consumer always demand what are these new technologies? Actually, these are the new technologies which does not require any temperature, because what we are aware most of the time, it is the temperature which preserves the food, and what we are talking about where there's no temperature is used. So there are several technologies. We'll talk about one or two technologies today because maybe time is limited time, and because for many years we have seen that thermal processing like canning or dehydration was the technology for the preservation of the food. And disadvantages of technical processing of the thermal processing is like flavor and the loss of nutrients. And physicochemical properties are affected most, like because of the temperature. So we'll be talking about new technologies here. And like suppose we are developing a new technology, most important thing is that there should be minimum losses of the flavor and the quality of the foods. It has to be maintained. Low processing temperature should be there because that will retain to most of the nutrients, and it should be at lower cost, and it should not affect the environment so much. It should be superior to the traditional technologies, and it should avoid reduces the detrimental changes like organoleptic properties should not be affected most here, and it should lead to the inactivation of the microorganisms and the enzyme, because these are the two factors which lead to the spoilage of the food when we preserve either the microorganisms or the enzymes. And we need to examine all that any technology, developed technology to see that how these microorganisms and enzymes are inactivated. So there are several technologies like high pressure, pulse electric field, ohmic heating, ultrasound, supercritical, and so many other technologies in the back of the card if you see. So we'll be talking about we'll start with the high pressure technology. If it time permits, we'll go to the pulse electric field, and all other technologies may be difficult to cover at the moment. So we'll just talk about the high pressure technology first. So it started with only the three major giants like Dan Farkas, Dallas Hoover, and Dietrich Knorr. They were met in a breakfast meeting in Dallas, and then they were thinking, when high temperature can inactivate the food, there should be a possibility that high pressure can preserve the food. And these people met in a meeting, and then they decided to build up a high pressure processing system. And it was made in US, and I was lucky enough to work in the laboratory of Dietrich Knorr for 2 years in Germany, in Berlin. And how the high pressure works here, it is very simple here, like high pressure kills microorganisms and preserve the food. It was known very well in advance, like 1899. And actually this is the high pressure. It was used by the ceramic industry for compacting the ceramic products. And later it was tried to extend it to the food processing. But it is something like this here. There is a unit here. Inside we put the food, and this plunger goes inside, and the water is pumped from the bottom. It creates a pressure all around the food. Only thing, we need to pack the food, and then we apply the pressure from all the sides. And then it is a hydrostatic pressure. It is not a gaseous pressure. So how high is the pressure, that we'll have to see. And it was very long, though it was invented in 1899, and food technologists offer in very recently in 1980, I can say, maybe 30 years before, that high pressure has many things to offer to the food industry. It was acknowledged very late. And Japan was the first company who launched, there was a company named Media Company, launched a high pressure process jam, jellies, rice cakes, and many other products, fruit juices in Japan. And later, these products have been launched in many other countries, like in UK, USA, Spain. All these countries have launched several products depending upon the traditional to the modern foods. And high pressure can at least, if it will be over exaggeration, if I say that high pressure technology can replace whole thermal processing. So rather, I want to be modest and say that, okay, it can at least supplement the conventional thermal processing to some extent, because it is going to be little more expensive than the thermal processing, because thermal processing is the cheapest one. What are the disadvantages of conventional thermal processing? Like, if we want to study the advantages of high pressure processing, we need to study the disadvantages of the conventional thermal processing. Like, canning is there. Canning is very difficult in the sense we need to penetrate the heat inside the food. Like, heat has to be penetrated, and then there is a hot point, a cold point determination, and then only the canning process time is calculated. And there will be a great thermal damage. Why? Because canned product will have a solid food and the covering liquid, and heat has to pass from the covering liquid to the solid food, and the mode of heat transfer is convection, and the convection from the liquid food to the solid, and from solid to inside solid, it's conduction. So wherever there is a temperature gradients, it leads to the lot of quality damage, and there will be a loss of volatiles. Color will be lost, flavors will be lost, and there will be a presence of unavoidable aspects of the chemical additives. Like, we don't use any chemical additives in the new technologies. So this is the disadvantages, and it will be all overcome in case of the high pressure processing. So we are just discussing how high the pressure is, like what is the range of the pressure in this. If you look at this, atmospheric pressure is 0.1 megapascal. So it is, what we are talking about is 100 megapascal, means 1000 times higher than the it is, hello sir, I am in a lecture. I will call you a little later. I will call you a little later. Okay sir, thank you. So how high is the pressure? It starts with 100 megapascal to 1000 megapascal, and it is something like that when two elephants stand one over the other on a 1 centimeter square cross section. The kind of the pressure generated will be around 1000 megapascal.
Sir, you are in muted, I think.
Sir, not audible, sir.
Some technical issue, madam. Just now I will find out which sir.
Okay. You call him, Sirish.
Ah, yes, madam.
Can you hear now? Okay. Yeah, I think it is solved now. I'll continue. Okay, so
Sure, sir.
okay, thank you. So how the microorganisms are inactivated in high pressure? It is because of the permeabilization of the cell membranes, and these changes are reversible at low pressure and irreversible at higher pressures. So this is like we need to apply higher pressures more than 400 megapascal, so it becomes irreversible. Otherwise, microorganisms will repair themselves and grow again. So we need higher pressures for the preservation. And for enzyme activity, like reduction of the enzyme activity ensures high quality and shelf life. These are the quality degrading enzymes like PPO, POD, LOX, and other enzymes which degrade the quality of the food. And why, how it is happening? Because when you subject the food to the high pressure, it leads to the unfolding of the protein chains. And basically the enzymes are the proteins, and it loses its catalytic activity, and it loses its activity in the pressure when you apply the high pressure. And another thing is here, when only the non-covalent bonds are affected. So most of the organoleptic properties like color, flavor are associated with the covalent bonds. So these things are not affected, color, flavor, and organoleptic properties, because it won't, only non-covalent bonds are affected. Covalent bonds are not affected. So this is we can apply a combination of the pressure and temperature method for Baroresistive microorganisms, bacterial spores can be inactivated if you apply, it cannot be activated, but if you apply pressure and temperature, both the like at a moderate temperature with pressure, then it is possible to inactivate microorganisms at this one. And there are not effect on the aromatic or sensory characteristics of the product. So what are the advantages? Like here, like we have a problem with the canning in that, because we need to cut the fruit, cover the water aqueous solution so that heat can penetrate. But here in this case, we are applying a pressure, and pressure is very uniformly distributed, like, and leading to very uniform quality, because pressure is uniformly distributed, is very instantaneous. These are the like a lead Chateliers and the isometric principles of the pressure, where pressure is instantly transmitted. There is no time delay. Whereas in the thermal processing, we come across the thermal gradients because temperature is not uniform, but whereas pressure is uniform. So whenever the temperature gradients come, this leads to the quality degradation. And there is an elimination or reduction because we are not applying any heat. So there is no degradation taking place because of the thermal energy here, and we don't use any chemical additives here. And sometimes it was seen that these kind of high pressure process products will lead to the generation of the new functional properties. Like it can means if you put a raw egg inside the high pressure, it will form a high pressure induced gel, and it is going to be very soft and much softer than the high temperature induced gel like omelette or something what we are making. So this kind of products are the new generation products and having a higher retention of the nutrients, which we have not seen. Maybe the coming generation will be using such technologies and making a new products. And it is effect, as we have discussed, it is because of the membrane permeabilization. It leads to the inactivation of the microorganisms. It can improve the overall quality of the food. Like when it is happening when there is no change in the temperature, the quality of the food will be improved, and it influences the phase transition. Like with the high pressure, we can generate 6 different types of ice. So if you look at this, we can generate a different kind of ice if we are making the ice cream. And ice cream, the taste, everything, raw material of the ice cream, anywhere you make it is same, but the kind of the way the ice crystals are formed, it is different. So the making ice crystals, we can generate 6 kind of ice. You must have seen snow ice, glass ice, and there are, and with the different densities we can make, and it gives a different flavor to the product. The different kinds of ice creams already coming in the market nowadays. So what are the other advantages? Like we don't have to cut the fruit into pieces like what we are doing in the canning, because we need to put, because smaller the size, better is the heat penetration canning. But in this case, is the pressure is transmitting, size is unimportant. Geometry of the sample is not important, but only thing is here, suppose we have a big fruit, we cannot keep in the high pressure because size, diameter of the high pressure is limited. So it is limited, higher the diameter, higher will be the cost of the processing system. So size reduction is not at all important for high pressure processing. Whatever the size we can keep, but only thing we need to care about what is the size available with us. And then there is a process flexibility and revolutionized food processing by making requirement of size reduction becomes obsolete here, because it was very primary operation canning. And here the possibility of making that at ambient temperature, we can sterilize the food, even pasteurize the food, or even at lower temperature, even at the sub-zero temperature. So if there is no other technology which can pasteurize the ice cream at temperature below 0. So high pressure technology, we can even pasteurize the food products below 0 temperature also. And it is basically, there is no waste generated, environmental friendly and energy efficient, in the sense, once we achieve the pressure, it is going to last for hours together. There is no change in the high pressure afterwards. So what are the application areas where we can use the high pressure? We can use the very lower pressures are required for the if we go for freezing and thawing, like we can freeze the product by application of the pressure, it will give us a different kinds of the ice. So we don't need a very high pressure. We don't need a very high pressures, like up to 300 megapascal we need, and temperature is around 10 degree. We can freeze and thaw the product, because thawing is a very slow operation. If we do at very high at the application of the pressure, it can be increased and we can reduce the drip loss. The product quality will be much better, because most of the time what happens, the loss of the quality takes place at the time of the thawing. So we can, high pressure thawing will reduce the quality of the thawed product, and we can do the pressurization, pasteurization, the blanching can be done, because generally if you are going for the dehydration of the fruits and vegetables, generally we blanch the fruits or vegetables putting in the hot water. That leads to the loss of the nutrients, and the blanched water disposal is a problem. Whereas in case of high pressure, we don't need any water. We can blanch the food just by applying a pressure. But purpose of blanching is to inactivate the enzymes like catalase and peroxidase that can be inactivated by the application of the pressure, and then it can be preserved, and it can go for the further process either of the dehydration or whatever process, canning or wherever. Whereas another process which has come up is the thermal sterilization with the pressure. It is called as a pressure assisted thermal sterilization, in which we apply a small amount of pressure, whatever means pressure plus temperature together for a shorter time, and it is similar to the canning or whatever thermal processing, but only thing, it is pressure assisted thermal processing, where we have added the pressure for a shorter time also in that case. So, but the pressure requirement is little bit higher. It may increase from 500 to 700, but only thing is, most of the time for canning, and we are going up to 121, but here we may not require to go up to 121. We can go up to 110 or 15. That is sufficient to reach the sterilization process. So just compare with the pressure assisted thermal processing with the high pressure sterilization. If you look at red color line is the retard sterilization, time and temperature history, and blue color is line high pressure sterilization. So look at this here, the lethality under the curve, it tells about the area under the curve tells about the lethality. See, the retard sterilization is so lethal. What is the energy gone into the product? But if you look at high pressure sterilization, it is not very high. It heats up very quickly. It goes up to 110. It stays there for some time, and then it goes down very quickly. So the preservation is because we are not spending too much time in the retard or high pressure. So the quality of the product is much better than the retard processed products. So this is the quality of the foods, like products we can make. The vegetables, these are preserved at ambient conditions and then does not require a refrigeration, and this quality is like a salad quality, very crisp texture, and not, because if you compare it with the canned product, sometimes canning it is very difficult to identify the shapes, but its shape, everything is maintained, and even if the microstructures are also maintained in case of the high pressure processed products. So this just to show you what are the different products and who are the manufacturers and the different countries. If you look at the first here for Japanese is the media was the first company launched jam, juices, yogurt, jelly. After that, there was so many other companies, like they worked with the tropical fruits and sometimes beef. And then USA, they worked with the guacamole and like dips, steady meals, fruit juices. And then US also worked on the meat and meat products, and then worked on the other products like poultry products, oysters. This is the best technology for oysters because it is there in the shell, and once you apply high pressure, it the inside that insect inside the shell comes out of its own, and it inactivates, it enhances the microbial status, like whatever the viruses or those things, it can inactivate. And in the other countries like France, if you see, many of the people are concentrating in the fruit juices, like apple fruit juice, other fruit juices, and some other like Spain and UK also worked on the fruit juices, and some are worked on ham and tapas, because this is their staple food, and these people have mostly worked. Most of the researchers in the world have been done on the either on the red wine or the cheese, because it is their staple products. So these are some of the other examples. Like this is from Japan. It is rice cake. This is called like guacamole. It's like avocado paste, and this is a very popular Mexican like a paste which they relish with almost with all the dishes. So, and it is not possible to preserve it with the thermal processing because it has a very sensitive flavors. And with this, whereas it can be preserved with the high pressure processing and can stay for a longer time. So these are some of the products like which is available in the national market. Like we can preserve the salsa, we can preserve uh, this one orange juice, and some other food products. Cut fruits also can be preserved, and there are many available. Even some of the Indian dishes made in US, people are selling, and high pressure processed products are available for Indian consumers. This is something like a food smoothie with different fruit juices mixed together like fruit punches. They are available in the market. Salsa. This is mustard sauce that is also present in many of the countries are selling this. And just to give you a feel that it is already commercialized. So various applications, like if you look at this, what are the different applications in the food industry? See, we can preserve the orange fruit juices, like in the bottle, we are getting orange fruit juice, but most of the time it is heated and then it is preserved and it develops some kind of a bitterness in orange fruit juice. But whereas if we pressurize only for 1 minute, it gives a very good shelf life of 12 weeks at low temperature. And then grapefruit, it is a very different fruit here. It has a peculiar flavor, and there is very few technologies which can preserve the grapefruit juice. Whereas in this high pressure technology, we can preserve the grapefruit juice, and this and lemon juice also. No fungus was determined in high pressure treated lemon juice. So because it has to be preserved lemon juice because and high end with the maximum retention of vitamin C. There was no loss of vitamin C and no changes constituents of the physicochemical properties. So pineapple can also be preserved, at least the fresh cut pineapples we can preserve as such, and pineapple juice also can be preserved by applying the high pressure. And mango is another product is the mango pulp and slices. Mango, India exports a lot of mango pulp to the other foreign countries. So basically it is based on the technology, it's a hot and filled technology. Whereas if we can use the high pressure technology, we just have to pressurize at around 500 megapascal just for 5 minutes and pack in the flexible pouches and export it. And this shelf life is around 1 month at 3 degrees centigrade. And when it reaches to the other different foreign countries, it can be opened and can be used either for maybe for the wine making or flavoring or anywhere it can be used. So it is the best technology for the mango processing. Guava is another fruit which is very sensitive to the flavor. If you heat it, you will lose all the guava smell. If you keep in refrigerator also, you will lose all the smell of the guava. There is no flavor. So high pressure technology can be used and we can preserve this guava puree for around 40 days at 4 degrees centigrade. So most of the time high pressure products are saved at least 3 degrees centigrade or 4 degrees centigrade. Why we are saving at lower temperature is here because this is suppose is there any chance of repair of the microorganisms, it may regrow again. So we subject it to a very low pressure, low temperature at the storage conditions so that there's no chance of reoccurrence. So avocado puree, we have just told it is called as a guacamole in the Mexican Spanish countries, and the peaches can be very sensitive taste, very sensitive flavor, which can be high pressure can be used for the preservation of the peaches. It leads to the inactivation of the PPO and other PPO, POD enzymes, and it is alternative to the blanching. We don't have to, if you blanch the fruit, then there is a problem because blanching will lead to the loss of volatile compounds. So we don't have to blanch the peaches. and then we can do high pressure blanching. And similar way, lychee is our fruit of India, and it is lot of work has been done on the lychee by the different countries also. We can inactivate the PPO and POD of lychee by application of the high pressure. And when these enzymes are inactivated, then we can preserve lychee in any way. We can preserve lychee as fresh. We can preserve lychee in the juices or sugar juices. Strawberry and black purees, like these are the foods which have a maximum antioxidant activity. Blackberry and strawberry. So like that, if you subject it to high pressure, it will lead to the inactivation of the enzyme and preservation of the food with minimum degradation of the food quality. And there is no significant change in the ascorbic acid, anthocyanin content, antioxidant activity. Color change is very less and redness was retained. Otherwise, if anthocyanin is there, if we heat this, anthocyanin is not susceptible to heating. So it is going to degrade. So in a similar way for the grape by red or white, it is generally used for making wine. And India grows a lot of grapes, and which is sometimes it is very good for the wine making. But how do we preserve this and send it to different countries who make the wine? Because French wine is the most popular because of the weather conditions and the grape, the kind of the grape they grow, and it can be sterilized with high pressure, can be kept for several months, and then whenever we want to make a wine, we can make a wine with the high antioxidant activity and anthocyanin. The color of the wine will be very red because it will lead to the preservation of the anthocyanin. And very interesting application of the high pressure is like we can keep the sugar, pectin, citric acid, and fruit juice, mix everything together, and then degas, and then we can process the high pressure, and then the jam will be ready, something like this. Without heating, jam is preserved. It is prepared without, and what we can do is we can even put a small fruit pieces here inside the fruit, the jam, because which we have not at least encountered such kind of products we have not seen, because it will give you additional extra taste of the exotic food products. Chunks can be put here, and these products are available in the Japanese market, but still come to India. So something like carrots can be preserved by using pressure state thermal processing. Tomato juice can be preserved by pressure state thermal processing. Green beans can be preserved because their base is susceptible. The color of the beans is very susceptible to thermal processing. If we do pressure state thermal processing, it is still green, and then we can keep it at just outside here. This need not to be kept inside the refrigerator at low temperature. Green peas is a big business in India as well as abroad, whereas we can preserve the peas by using the high pressure technology, pressure step thermal processing, and then it can be still available as a fresh. We can preserve the mushroom also using the high pressure technology, and there will be less texture degradation. Mushroom is very important for the maintenance of the texture, so texture will not be degraded so much, and browning can be reduced because tyrosine or whatever enzyme present in that will also get inactivated because of high pressure. And if we evacuate, like remove the air from the sample and then apply high pressure, it will lead to the extended shelf life. Only thing is we need a very little bit higher pressures required for the mushroom. It has many advantages to offer to the dairy industry. Like if you apply high pressure, it can extend the shelf life of the milk even. In combination with the high pressure or the temperature, we can increase the shelf life. And if we obtain, it will have a synergistic effect when high pressure combined with the antibiotic, bacteriocin, antimicrobial peptides we can combine, and then it gives us an additional shelf life. And it has lot of advantages in the cheese making. Like cheese, when you make the cheese, it has to be rennet. We need to add a rennet coagulate, and then protein has to be coagulated. So it is reduction in the rennet coagulation time. When we add a rennet for the coagulation and we high pressurize this like that, what will happen? It will reduce the time of the cheese making. And the biggest advantage, and it will reduce the time rate of the curd formation. So also will reduce. And biggest advantage, it will increase the yield of the cheese. How it is increasing is like when we apply high pressure, there are proteins which are going waste in the way as a way here. that will also get coagulated, and then it will increase the yield of the cheese, and then that will increase the water holding capacity and denaturation of the pea protein association with the casein, and all the things will happen. It will increase the yield of the cheese. Like earlier, suppose 1 liter we are getting 100 grams, now we are getting 120 grams. So 20 increase of the cheese is increased, and it is ripening. If you say cheese ripening, that also it will increase, like it will lead to the proteolysis of the milk protein, and this led to the increase of the free amino acids will increase, and that it will lead to the improvement in the taste, because the kind of the cheese which is sold in the foreign market is very different what we get in India, because this proteins has to be broken into the free amino acid content, and then leads to the improvement in the cheese taste. Yogurt, another milk product. When you make a yogurt, there is no technology which can which can stop the growth of the microorganisms, leading to the spoilage of the yogurt. So we can apply high pressure technology for yogurt, and once it reaches to a particular acidity, we can stop it. Only thing we need to have an optimization in such a way that this whatever the bacteria required for the health should not be degraded here, and only thing we should stop the bacteria which is which is required for the acid production. It has some advantages for the beef. Somehow it is not very good technology for meat, because if you see that red color, if you apply at different pressures, it will become little bit white. So there is a little change, but it will lead to the extension of shelf life, and it is leading to the oxidation of the myoglobin to ferrous microbin to ferric myoglobin, and the color to gray brown. That's why you are seeing red to it will become gray brown. This is not a very positive sign, but anyway, it will improve the microbial status of the meat, beef. Enhancement of water holding capacity will increase. Texture, consumer acceptability will increase. Sensory quality is very high, but only thing is color. We need to look into it, how to improve the color. Like fish is if you are preserving fish by using high pressure technology, it will lead to the reduction in the total plate count, increase in the freshness index. Like there are so many other parameters which are all going in the favor of the fish, and it will like TBHQ or whatever we are thinking, it will reduce. Like it will give an extended shelf life to the 160 to 180 days. Like 2 months to 6 months, it can increase the shelf life at lower temperature without significant chemical, microbial, or sensory changes. So oysters we are talking. This is generally oysters comes in the cell, and then when we apply high pressure technology, it leads to the taking out, inactivation of the pathogens. It will inactivate detachment of the muscles, retention of good shape, and more voluminous and juicy. So it is very good technology. That's why there are so many companies in US. They are using this technology for the oyster taking out from the shell, because it will detach automatically. It will make a good shape and makes a more voluminous. It can be even used for the chicken, duck, and turkey also. Same way, like that, it can be, if 5 log cycles reduction is possible in the Clostridium sporogens, then it can be considered as a preservation technology. So it can be coupled with the high temperature and low storage temperature, then it can be considered as a potential technology for extension of storage life up to 98 days. It can around more than 3 months it can extend. So it can be even used for these are the meat products like sausages also. It can increase the shelf life, and egg yolk and egg white, because this is another trend which is coming up. People in the West, they don't want to break the egg and make the cake. So what they are going to do is, like they will buy the egg yolk and egg white separately, and whatever required quantity is there, they will make a cake out of it. But how to preserve this? Because egg yolk and egg white, moment you heat it, it is going to coagulate. So high pressure, optimized high pressure technology may be very useful to retain its original color, softness, and adhesiveness. And anyway, thank you very much for your attention. And it is a very promising technology which has come up in many ways, and probably it is going to hit in India very soon. If it is not going to hit in India very soon, we will get a products made in China and coming to India. So in order to stop that, many of the big giants are probably, I think, they will start the production of the high pressure process products. Some have already started. Few products like the natural sugarcane juice and the coconut water is already available in the made by some of the companies, but they are not declaring as high pressure process products. So maybe in future it is going to hit in our country, and somebody takes initiative, it is going to happen. Thank you very much for your attention, and I'll stop it here.
Thank you, sir. Now the session is open for queries, ma'am. Any doubts or discussions?
Sir, you have mentioned for tomato juice also.
Yes.
This technology. So how many days we can store this tomato juice, sir?
See, if you have a pressure assisted thermal processing, actually it is the combination of the pressure and temperature. Those products can be retained even for more than a year. So generally, yes, it is because what we have a technology like for the tomato processing is that just hot filling is sufficient. Like in many of the natural calamities, CFTI is distributing food products like a chapatis and the tomato chutney. They require only just hot filling because it is consumed immediately within 6 to 7 days. If it requires for larger time, we can use this technology. But only thing is that it is in the stage of the development now, and it is approved by US FDA. And US FDA I worked in Ohio State University, so US FDA has used some of our data to validate this technology and given approval for this technology even for the pharmaceutical and other products. Slowly it is coming for the food technology also, because problem is coming with the bigger size food processing equipments, commercial scale.
How much it will cost, sir? For
cost is little bit. It should not deter people to use because capex is very high for this. Because if you go for a bigger unit like around 50-55 liters, which is available at NC Hypervedic US, that is around maybe 5 to 6 crores. And that is the industrial unit. And if the operation is very for 5 minutes or 2 minutes like that, the operating time and operating cost is very low. So even if you have a smallest one equipment, you can speed up the production by reducing the time.
Okay, thank you, sir.
Sir, at present in our country, in this high pressure technology processed products are available.
Yeah, there is a company which claims that they are making a high pressure processed products. There is a company in Bombay, like their raw pressery. Their website is also available. But when I found the products are available in the airports and the bigger canteens like Infosys and all, there is no mention about if they are high pressure processed products. Because I don't know what is the reason for that, because they never consulted CFTRI or they didn't come here. But I came to know that they are making such products. But if there could be a better quality improvements can be seen if they really make a high pressure process product. I am thinking this is a hot filled product and they are not mentioning anywhere it is high pressure process.
Okay, sir. What about this pulsed electric field processed one, sir?
Yeah, it is another with the non-thermal processing technique. In the pulse electric field, we give an electric field in the form of the pulses, and this is like it will not lead to the heating up of the product. Like milk can be processed. So if you calculate the amount of the heat transfer to the food, it can heat not even more than 1 degree centigrade. So that's why it was in the continuation, but looking at the timing, I thought it's better we talk only about this because don't want to create a mess.
In future, we will plan another.
Sure, anyway, now we have developed the relationship, so we can come and talk to you about anytime we can. I'm taking up many of the classes. I'm a chemical engineer, but taking classes for the emerging technologies for food processing, post-harvest technology, and whatever comes in the way, we can teach. And I met your students who are here.
Thank you, sir.
Okay, ma'am. Thank you very much.
Any questions, ma? Okay, shall we conclude, madam?
Yes, ma. Yes.
Now I welcome Beulah conclude the session. No?
Viola mute lo unnaru ma.
Yes, ma'am. Just now I am allowed unmute, ma'am. Thank you, Dr. Naveen Kumar Rastogi, sir. Thank you so much for giving very interesting talk on topic emerging food processing technology for better retention of nutrients. Sir, you have clearly mentioned about the high processing preservation technology, which helps in keeping the fresh produce with acceptable and convenient shelf life, with assuring safety and nutritional value. And it also helps in the inactivation of the oxidative enzymes and food pathogens. At present scenario, maintaining the fresh appearance characteristics along with minimal processes without losing the quality plays a crucial load, especially in foods like fruits and vegetables, dairy and meat food products. But here with the usage of high pressure processing, preservation processing method, we will have a better stability of minimize the loss of viable ascorbic acid and other nutrients when we compare with the thermal processed ones. And also this will help in maintaining the all characteristics and will be a supportive to develop a new food products by the food industries, and will also help in minimizing the wastage of food. Thank you very much, sir, for the excellent and informative presentation.
Thank you, sir. Further, we will collaborate in different activities. Just.
It is our pleasure, ma'am.
Thank you, sir.
Thank you.
In our university, we have a 5-year integrated food technology course. So it is in the establishing stage. So we need your help for further.
Yeah. Your student, Shivani Tripathi was here. She wanted to do PhD here.
Yes, sir. Yes, sir.
So we are just guiding her. I said you contact madam and come back to us, which is your interest.
Yes, sir. Yesterday night she messaged me.
Okay.
Thank you, sir.
Thank you, ma'am.
Now we are closing this session. After lunch by 2 o'clock, technical paper presentation sessions will be started exactly by 2 p.m. All of you check your schedule, given schedule. According to that schedule, please join that technical session. So here 2 parallel sessions are going for the presentations. So all of you join the sessions and complete your presentations. Thank you.
Hello? Shirisha?
Madam.
Will there be any 2 different Zoom links or the same link?
This Zoom link is for 1, 3, and 5, madam. Another Zoom link is provided for session 2, 4, and 6.
Okay, thank you.
Thank you, ma'am.
Okay.